May I introduce the meeting? Thank you, everybody, for the participation uh, to this meeting, and I would like to introduce you the garden project for people that don't know what it is. Garden is a global hematological rare disease alliance that is rooted on the foundation Franco and Piera Cutino. And the main aim of this network is to create a collaboration between opinion leaders in different hematological rare disease. This is the peculiar characteristic of Garden network among people who work in different rare hematological disease, and even to have a network between among the patient association of the patients and among the patients to the opinion leader. This is because we think that hematological rare diseases are different, but the concern about accessibility and sustainability of innovative drug is the same. So we are promoting collaboration and innovation, and this is one of the steps of this project that is training, to make training to increase awareness about this disease, how it is possible to overtake a barrier on accessibility and sustainability of these innovative drugs that is the same in different hematological rare disease. Where we are today, we started this year with a kickoff meeting with different opinion leader, 45 opinion leader from 19 different countries. And we have another webinar in September on advanced treatment in rare hematological disease. And now we are celebrating the first meeting in collaboration with a regulatory agency, with HIFA. We have two representatives of HIFA today, Professor Paolo Foggi, Professor Antonella Esgro, and this is another step of Garden Network that like to work together with regulatory agency, and we hope to organize another meeting with other European regulatory agency. So this is our aim, and I would like to thank everybody for participating to this webinar in a day that is Saturday, that normally dedicated to the family. Now I think that I move. We can move with the first session with the chair of Professor Messori.
Thank you, Aurelio, for inviting me. And so this is the first session. It deals with thalassemia, hemophilia, and anemia. And as you can see from the program, we have 4 speakers. And I chair this session because Professor Rivella could not attend this meeting live, and so I am here instead of him. I think that we can start directly by introducing Professor Rivella, whose presentation is a pre-recording video, and we can start with this video, please.
Hi, everybody. I'm Stefano Rivella, and I want to thank you for inviting me here and give me a chance to share some of the data we generate in my lab. And I want to thank in particular Dr. Maggio and the Fondazione Franco e Piera Cutino for this opportunity. So today I'm going to talk about our work in the field of gene therapy and how we move from establishing mouse model and testing novel gene therapy tools to potentially novel therapies and including a clinical trial. So let me say that the first thing is I want to share my disclosure. But for the aim of this talk, I just want to make clarify that none of the data that we'll present today have been supported by industry, but all of them by the FDA, the NIH, the FDA, and eventually the internal grants through CHOP, Children's Hospital of Philadelphia. So let's start from the difference between, when we talk about gene therapy, the difference between application that are indicated at ex vivo versus in vivo gene therapy. So in the more classical and present therapies, gene therapies are defined ex vivo because everything in terms of modification of the hematopoietic stem cells has to be done outside of the body of the patient. So what does it mean? It means we need to basically mobilize the hematopoietic stem cells that we indicate as a CD34 positive cells. And this mobilization is followed by apheresis, where these cells are collected outside the body of the patient, where eventually they are manipulated by, for instance, lentiviral vector that are important for the technology indicated as a gene addition or gene complementation, or more recently by additional tools that basically modify the genome of the patient by CRISPR-Cas9 based tool. So once the cells are genetically modified, they need to be reintroduced back into the bone marrow of the patient. However, this requires myeloablation, which is basically a toxic approach, a toxic treatment to eliminate the stem cells are still present in the bone marrow. They're still carrying basically the mutation. In the case of thalassemia, one of the mutations lead to the decreased expression of the beta globin gene, while in sickle cell anemia, the single mutation that leads to the polymerization of the sickle cell hemoglobin. And so you need to eliminate these cells, but also you need to make space for this genetically modified hematopoietic stem cells that potentially can cure these patients. So in the most recent approaches that have been at this stage only presented or studied in animal models, there is potentially a new way to do gene therapy. It's called in vivo gene therapy, where the tools that are devised to basically modify genetically the hematopoietic stem cells are infused directly in circulation. And these tools could be lipid nanoparticles, could be virus-like particles, for instance, or directly viral particles that can be infused directly in circulation and with the goal to target the hematopoietic stem cells in the bone marrow, so that the genetic modification is done directly in the bone marrow and with preserving or eliminating the need of doing the so-called myeloablation. So the stem cells don't need to be taken off and modified ex vivo, because these tools theoretically potentially can target directly stem cells in vivo. So in the field of hemoglobinopathies and gene therapy, there are now the gene therapy is now a reality. There are basically gene complementation strategies under the name of Zynteglo Lyfgenia, which are based on lentiviral transduction of hematopoietic stem cells with a virus that carries the human beta globin gene. And these are available for both patients with thalassemia and sickle cell anemia. However, these approaches generally require high vector copy numbers, so a large number of integration in the genome to be curative. In addition, there is also a new approach based on genome editing, which is CRISPR-Cas9 technology, that basically targeting the BCL11A erythroid enhancer prevents the expression of this gene, which is a repressor of the fetal hemoglobin, reactivating this potential curative hemoglobin that can definitely improve or cure patients affected by both beta thalassemia and sickle cell anemia. However, the treatments for these diseases are very costly, and so in my lab for a few years, we're trying to address the following questions. Can we generate a novel vector that is intrinsically safer, effective, and more affordable than beta thalassemia and sickle cell disease? And can we also develop a similar gene therapy approach for alpha thalassemia? Since we're talking about hemoglobinopathies, I want to share that with you the information that this similar approach can be utilized also for this additional form of hemoglobinopathies. So I'm going to show data that been published now a few years ago. The person that led this project was Laura Breda in my laboratory. And so a few years ago, when we were already at the Children's Hospital of Philadelphia, we decided to make a novel lentiviral vector. But we didn't want just make a lentiviral vector and expecting this to work well by itself, but we want to also compare this to lentiviral vectors at that time were currently available or proposed for clinical trials. So we made a number of novel lentiviral vector indicated here in this area in orange, where basically we included a human beta globin gene and different elements that control the expression of the beta globin gene. And we also included an anchoring insulator for safety and additional modification to increase the titer and the production of this lentiviral vector. Then we selected one vector we call LS20, and in the following studies, basically we compare LS20 with additional vectors such as GLOBE, TNS9, and BB305. For instance, BB305 is the vector utilized by Bluebird under the name of Lyfgenia Zynteglo, and we compare our vector to this vector using a variety of tools such as cell lines, stem cells from patients, but also mouse model. And at the end of the story, we observed that our new vector LS20 was producing more beta globin than these other vectors in the range of 40 to 150% more beta globin. And with this data, we asked permission to the FDA to treat now patients affected by beta thalassemia. And eventually, if the trial goes well, we'll also include patients of beta by sickle cell anemia. So our clinical trials is currently is open, and we're treating the first patient, and we hope to have good news in the next few months. So, but the beta globin gene vector also open for us an avenue to treat alpha thalassemia. So to do this, we basically establish a new mouse model of alpha thalassemia major What does it mean? These are animals that basically don't express the alpha globin gene in their stem cells. We can delete this gene by a novel technology and defined as a lipid nanoparticle delivery mRNA tool. And basically, with this technology, we can eliminate the alpha globin gene in the bone marrow of this patient. And when this is done, we can use this bone marrow, analyze the mice, or use this bone marrow for transplantation in normal mice. And what we observe after in mice that basically don't express any alpha globin gene, we observe that these animals affected by very severe alpha thalassemia, where there is no expression of the alpha globin gene, there is splenomegaly, there is iron overload, there is a lot of production of erythrocytes in circulation, and this is due to the fact that these animals make only hemoglobin H, which is basically a beta globin type tetramer. And this hemoglobin H binds oxygen at very high affinity, but it doesn't release oxygen in circulation. So the mice are extremely hypoxic. And for that reason, they make a high level of erythropoietin that basically feeds on the red cell production, even if these red cells are completely abnormal and non-functional. And they also lead to a high level of VEGF and other characteristics such as microvascular occlusion that was a very interesting feature of this mouse model alpha thalassemia. But then what we did, basically, we modified the LS20 vector that originally carried the beta globin gene, and we modified this vector with replacing the beta globin gene with the alpha globin gene. And using this vector now, we can basically introduce the human alpha globin gene in cells that are not, in mouse cells that don't express any alpha globin gene. And in doing this, basically, we were able to completely rescue the mice. One copy of this vector was sufficient, basically, to rescue the mice from alpha thalassemia. I didn't mention that the mice without alpha globin gene, from the moment that they basically stop expressing the alpha globin gene, in roughly 6 to 8 weeks, only make hemoglobin H and eventually die due to all these consequences, lack of oxygen delivery, and many other problems such as the microvascular occlusion. However, when we use this new vector we call LS20 alpha I, which is one of many vectors that we tested with the alpha globin gene, we're able to rescue these animals and showing that as a proof of principle, a single copy of this vector potentially can cure alpha thalassemia and potentially in all these forms, from the most severe one where there are no alpha globin gene expressed to the less severe forms with 1 or 2 copy of the alpha globin gene are present, but the patients are still affected by a relatively severe form of alpha thalassemia that may require blood transfusion. So all these new vectors have been developed under the umbrella of a center at CHOP we call QRED, that stands for the Comprehensive Center for the Cure of Sickle Cell Disease and Other Red Cell Disorders. This is a center that I established together with Janek Yakowski. She's the clinical director and my scientific director. And the idea here is basically to help patients to be treated at our center very rapidly in the sense that when they come for, let's say, a thalassemia patient comes for routine visit, they see all the doctors from the hematologist to the cardiologist, etc., the same day. But they also exposed to potential clinical trials. In our center, for instance, we was one of the center where Lyfgenia and Zynteglo and Casgevy were utilized in the clinical trial. And so we provide the patient access to this trial. And we provide also a number of other additional support, such as psychological support. And we discuss about fertility, how to preserve fertility, etc. And so this is a center that really try to help the patient from many, many aspects, not just from the curative aspect per se, but also for the treatment, the follow-up, and providing information at different levels. So, but I'm personally also very interested in establishing a new model for gene therapy, what I didn't say that the trial that we perform in our CHOP with the LS20, it hasn't been supported by any company. It's been done simply by collecting, as they would say, grants and also internal support. This is possible because CHOP has facility to make the lentiviral vector, has facility to do the mobilization, the pheresis has facility to do the transduction and the bone marrow transplantation. And so basically the goal is really to show that we can do a clinical trial in our center that potentially 10 times less costly than what normally now is utilized to treat patients with thalassemia with industry-related products. So basically, our goal is really to the QRAD framework as the goal is to advance an hospital-based nonprofit but sustainable model where hopefully we can share manufacturing know-how, open protocols, and also introduce this to other centers, hopefully, such as mirror trials, creating partnership where in order to cut costs and grow cohorts of patients while meeting local regulatory needs. So the plan also aims to standardize vector supply and data sharing, aligning this technology and GMP production in many centers, and in order to avoid duplication, accelerate access. And of course, this model specifically targets ultra-rare diseases that would otherwise go untreated, building a patient-centered value chain for advanced therapies. And I want to stress rare diseases because, as you can imagine, for instance, right now we have a vector for the treatment of X-linked sideroblastic anemia, and there are very few patients, and we don't think that many companies or all company or any companies will be interested in helping us to treat this patient because it's very difficult to make a profit by treating a few hundred patients. So, but I want to finish in the second part of my presentation. I want to talk about targeting hematopoietic stem cells in vivo. The advantages, already mentioned the advantages in one of my earlier slides. The idea is basically to correct the disease directly in the hematopoietic stem cells by targeting the mutation. This would, for instance, if we can do this by not using lentiviral vector, we could eliminate the random integration. And this approach may also help us to reduce or eliminate the toxic myeloablation. And I'll show you some preclinical data as a proof of principle that this technology can be utilized. And so this technology utilizes basically lipid nanoparticles. These, like, in this center part of the picture, you can see a lipid nanoparticle, which is basically a way to deliver mRNAs. And when you deliver mRNAs and you target hematopoietic stem cells, you can do multiple things. You can modify them genetically. You can change their behavior. And as a proof of principle, we show in this paper that was published in Science, again, the leading author was Laura Breda in my laboratory. We've been able to show that by this lipid nanoparticle, for instance, we can deliver an mRNA that expresses the luciferase gene so that the stem cells or the organs where the RNA is delivered became fluorescent to the green color. Or we can deliver a Cre mRNA that the function of this Cre mRNA, when it's translated, is to basically eliminate a region indicated by these two locks beside, such that this promoter can now express a marker gene which is fluorescent in the red color. And if this is working, so we achieve this deletion, then this basically gene is activated and the cells became red fluorescent. And I want to show you some data here, basically showing that if you do deliver this LNP mRNA expressing the luciferase gene, we can see activation of this protein in the liver, not only in the liver, but also in the bone marrow, which is where we would like this mRNA to go. And as a matter of fact, if you deliver this Cre mRNA, so that the cells, that basically the stem cells are genetically modified, the progenies became fluorescent for the red color, you can see basically that with the Cre mRNA that targets the bone marrow cells, we can achieve a very high targeting of cells in the periphery, such as red cells became fluorescent to the red color, but also white blood cells, or hematopoietic stem cells indicated as by the c-kit of LSK markers. I want to mention that in order to deliver this LNP to the bone marrow, in particular to the hematopoietic stem cells, we need to use an antibody that targets the c-kit receptor or CD117 receptor, which is expressed at high level on the hematopoietic stem cells. And so this is what happens if you use this LNP with this antibody that targets this receptor. Well, these basically are in this case, here in the lower part is a control IgG, where basically very few cells are targeted by an LNP that doesn't have the antibody. And here's just to show what happened if you use two different quantity of the this LNP. This is 1 microgram and this is 5 microgram. And you can see there is an increase in the target in the hematopoietic stem cells or red cells in circulation if you use obviously a higher amount of this product. I want to show you that we can also target hematopoietic stem cells at this stage still ex vivo. These are human hematopoietic stem cells. And we can target with RNAs that can modify the sequence of the beta globin gene or the BCL11A gene, which induces fetal hemoglobin. In this case, we use hematopoietic stem cell from a patient affected by sickle cell disease. This is the HPLC profile that basically shows the expression of the beta S, the secret beta S beta globin together with alpha globin. And after treating this hematopoietic stem cells with a gene base editor that modifies the beta S to beta Makassar, which is considered much safer and potentially curative beta globin chains, you can see that now we have a very large proportion of this hematopoietic stem cell that after differentiation can make this curative beta globin called beta Makassar indicated as a beta G. While a minority of this hemoglobin now is producing beta S. Similarly, if we can target, in this case, wild-type hematopoietic stem cells with a Cas9 CRISPR-Cas9 approach that targets and answers the BCL11A gene, you can see we can increase basically the fetal hemoglobin from basically very low levels to levels that would be considered curative. This is, for instance, is a normal patient, but this is a patient affected by sickle cell anemia. And you can see now that a lot of hemoglobin F is produced in these cells. Now we're basically testing these, as we speak, these tools with a novel formulation of LNP on hematopoietic stem cells in vivo. And we hope to have positive results in the following months. I just want to show you that we can also target, we can already deliver RNA to hematopoietic stem cells in vivo. This is, however, not the base editors that I showed you before. These are mice that basically xenograft, that have been basically transplanted with human hematopoietic stem cells. And with a single infusion of LNP that deliver ZS green fluorescent protein, RNA that basically translated can show fluorescence in these cells, you can see that we can reach an average 5% of targeting in these hematopoietic stem cells, human hematopoietic stem cells in mice. But we really hope to increase the targeting to levels that are considered more therapeutic in the range of 50% or higher, and eventually combine this with a base editor mRNAs. And as I said, these experiments are in progress, and we hope to see positive results in the next few months. And so future directions, the work done using mouse model of hemoglobinopathies can be further utilized to develop in vivo gene therapy strategies. So one thing that I didn't have time to show today, but we're also using this in vivo delivery of RNAs by lipid nanoparticles to basically eliminate hematopoietic stem cells. This would be an alternative and less toxic myeloablative regimen. And we have already data, we can do that while we are preserving the fertility. So the mice can go back after elimination of these hematopoietic stem cells and bone marrow transplantation. They can go back to their cage and can still breed. And then, of course, we are also generating humanized models of alpha thalassemia, other diseases, So in order to test directly these gene editing tools in mouse models or in xenograft models, with the goal hopefully to being able one day to use gene editing tools in vivo directly, and so eliminating completely the requirement for myeloablation.
Thank you, Professor Ribella. Are there any questions from the panelists? Otherwise, we can proceed. Now, actually, I see that the first presentation was
There's some problem with
from Professor Aurelio. And now we can invite Aurelio Maggio to give the first presentation of the program. And so, thank you, Aurelio. You can start.
Okay, thank you. I have to left this. Okay. Boo.
I stop your sharing, Professor Maggio. Try to share again with
Okay.
the correct file, please.
Okay. Oh, I have some problem with this. Okay. Okay. Sorry. Hold on. Okay.
Okay, perfect.
Oh, thank you. My talk is on reframing thalassemia syndrome as a benign hematopoietic stem cell disorder. I hope that I may convince you that this is a correct classification or definition of thalassemia syndrome. And the next.
Just click twice on the slide.
Okay,
Okay.
it is my conflict of interest, and we know that by some paper published by Stefano Rivella that um, what we know. Um, this paper suggests us that the variability of thalassemia phenotype severity is not related only with the imbalance of globin chain synthesis, but even is related with changes in erythropoiesis efficiency and in the iron metabolism. The second point is that we know, even by other paper published by Bart and Rivella, that in thalassemia we have a constitute activation of EPO receptor of STAT5 pathway by JAK2 JAK2 gene, and this is the same that it happened with in polycythemia vera. Finally, we know even by the real world treatment that we have now that we can use drug like luspatercept to inhibit growth differential factor 11, and then we have other mechanism of increasing hemoglobin like the inhibitor of IHIF2 alpha, and then this means that may influence the severity of anemia in patients with transfusion or non-transfusion dependent thalassemia in a different way. So what we done with Professor Napolitano and Alita R, was to review the literature in order to explore whether dysregulation of hematopoietic stem cell could be explained phenotypic severity in thalassemia. And at the start of this of this issue, we have to remember which is the complex cellular machinery regulating the fate from quiescent hematopoietic stem cell to active hematopoietic stem cell. This pathway is different. One is the cell cycle entry pathway. The other is the mitophagy or autophagy pathway, or metabolic activation pathway, or epigenetic modification pathway. Each of these pathways decide if the hematopoietic stem cell may move from quiescent to active state. And this is very important in the mechanism of differentiation and apoptosis of cell line in the bone marrow. If we look at this pathway, we see that cell cycle entry regulate the transition from the G0 to the G1 phase of the hematopoietic stem cell, and this is done through an antagonist mechanism. One is cyclin-dependent kinesis, and the other is the inhibitor of cyclin-dependent kinesis. One promote and other inhibit cell proliferation. Another mechanism is autophagy and selective mitophagy. The hematopoietic stem cell control the metabolism of the cell by mitophagy and by autophagy, and this is important because in this way the stem cell can have a quiescent state inhibiting phosphorylation metabolism. According in the other pathway is the metabolic activation, and this is crucial to maintain the quiescent state of the cell, and this is a mechanism that is based on the control of radical oxygen species. Finally, we examine, we can look at epigenetic modification. This is regulated by acetylation and deacetylation, by methylation and demethylation. And the mechanism is to control compactation or decompactation of chromatin. Each of these mechanisms, for example, acetylation and methylation, decompact the chromatin, while the acetylation and methylation compact chromatin, and this is important in the regulation of gene expression because control the entry of the cell factor. So if you examine what happened in thalassemia hematopoietic stem cell, we can see that for cell cycle entry, it is documented that it's shown that we have a reduced level of CDKS, cyclin-dependent kinesis, and we have an increase of inhibitor. And this increase of inhibitor of cyclin kinase are even regulated by an EPO-independent pathway. If you look at autophagy and selective mitophagy, we know that generally in the cell, the quality control of mitochondrial is regulated by three processes. One is the metabolizing of unfolded protein by AA protease complex. The second is to select mitochondrial protein to transfer this in vesicular and to transport to lysosome. as it's possible to see on this cartoon. And the other is the entire mitochondrial phagocytosis that is at the end transfer in a double membrane vesicle, and this is and transfer to a lysosome. And as it is possible to see in this cartoon, we have in thalassemia CD34 studied by cell culture, upregulation of gene involved in autophagy as ATG5 and LC3, and we have an increased transcription level of gene associated with erythroid mitophagy that are BNI, P3L, and NIX. So this means that ineffective erythropoiesis in thalassemia stem cell is not only due to the imbalance of beta globin gene, but even in the dysregulation of mitophagy and autophagy. Concerning metabolic activation, we have two mechanisms that are dysregulated. One is the NOx pathway, and the other is the overactivation of ERK1, 2, and the constitutive dysregulation of JAK2 phosphorylation, the same mechanism that happen in polycythemia vera. NOx control the oxygen consumption and so control the amount of radical oxygen species that are increased, but are increased even in leukemia myeloid acute cell, because the expression of NOx gene is a marker of acute myeloid leukemia blast differentiation. And we know that in thalassemia we have an increase of ROS level, and this means that even an NOx gene expression is dysregulated. Additionally, in thalassemia, we have a very low level of buffer protein as antioxidant because of iron overload. Concerning the mechanism, the second mechanism of metabolic activation, in thalassemia stem cell, we have overactivation of ERK1, ERK2, and a constitutive metabolic dysregulation of JAK2, as we already said. Epigenetic modification have been shown because we have an increase of CYST1 enzyme promoting acetylation. And this means that we have an inhibition of BCL11A, KLF1, and ADAC1. This means that this mechanism inhibit BCL11A. We have also an increase of expression of some microRNA. So, and finally, we have an involvement of mTORC1 through ULK1 mediated, this gene mediated autophagy, and this is involved in excess of alpha globin. And we know in vivo trial that, for example, sirolimus is able to inhibit this action of autophagy, and there is some publication of increase of hemoglobin with the use of sirolimus in patients with thalassemia. So we conclude from this evidence that we have the same dysregulation mechanism in hematopoietic, in thalassemia hematopoietic stem cell. Perhaps they are different in severity, but they are similar to that we have in malignancy, hematological malignancy disease. And to make another another issue on this, we can see that we already use some drugs that are used even in hematological malignancy. For example, hydroxycarbamide or luspatercept, working on cell cycle entry and are used in hematological malignancy and thalassemia. There are some drugs that are used in mitophagy for leukemia and are in progress to be regulated by FDA. And for metabolic activation, we have thalidomide that we use in some severe form of alloimmunization thalassemia patients or sirolimus. And for epigenetic regulation, we have the example of azacitidine that was used in sickle cell. We know that there are a lot of gene in this cartoon are you can see on yellow color that can be influenced BCL 11A, and that could be target for next future therapy. And other mechanisms are epicidine pathway of fibroblast growth factor 23. So we can see that thalassemia hematopoietic stem cell is dysregulated as other oncogenetic or hematological malignant disease. And finally, I would like to remember everybody how the name, the definition of thalassemia was coined. This was coined by, the name of thalassemia was coined by George Whipple and William Bradford in 1941. And this was coined, this name was called because the relationship between the disease and the provenance of the patients from Mediterranean region. However, if you look at Cooley, Thomas Benton Cooley, he described the thalassemia as erythroblastic anemia, and this is more close to the definition of a disease that could be included in disorder of hematopoietic stem cell. So, although additional research is required, I think that considering thalassemia as a benign hematological disease is more close to the phenotype of different phenotype of this disease, and open the development for use new innovative treatment that already are present in hematological malignancy. Thank you very much.
Thank you, Aurelio. And so we have the last session, which will manage your question and answers. And so we can directly proceed to the third speaker. This is a live presentation. And so I invite Oriana Marquez. Thank you for your participation. And you can start now.
So good morning, everyone. I would obviously like to start by thanking the Scientific Committee for the kind invitation. It is actually a truly honor to be part of such an important initiative to talk about molecular approaches for controlling our pathways in anemia. And I guess the initial question would be, why controlling iron during anemia, right? And the answer lies in this enormous demand for iron during erythropoiesis. And this happens predominantly in the later stages during erythroid differentiation and maturation because of the need to synthesize hemoglobin as part of the red blood cells' function to transport oxygen. And so considering that each hemoglobin molecule has 4 heme groups, with each heme group requiring 1 atom of iron, and that 2 million red blood cells are produced per second, This means that 20 to 25 milligrams of iron are needed daily to meet the requirements of erythropoiesis. And so, in fact, iron availability is a rate-limiting step for hemoglobin synthesis, with its deficiency resulting in anemia. So then how does the body control iron availability to meet this enormous demand? So when iron is needed, such as during increased erythropoietic demand, the liver responds by reducing hepcidin production. So hepcidin is a liver peptide hormone that controls systemic iron availability by binding to its iron exporter that is called ferroportin and triggering its degradation. So when hepcidin levels fall, ferroportin is preserved on cell surfaces, which has two important consequences. First, in the duodenum, dietary iron absorption increases because enterocytes can now export iron into circulation. And second, reticuloendothelial macrophages that recycle the iron from senescent red blood cells can release their iron source back into circulation. This iron circulation is rapidly bound by transferrin, which then delivers it to the rest of the body, including to erythroid cells in the bone marrow, enabling hemoglobin synthesis and red blood cell production. On the other hand, in conditions of iron overload or inflammation, hepcidin levels increase. And in these conditions, hepcidin, by promoting the degradation of the iron exporter ferroportin, blocks iron absorption in the gut and also iron release from reticuloendothelial macrophages. And so this creates hypophoremia or reduced serum iron levels, which, if prolonged over time, will affect erythropoiesis due to iron starvation. So it's easy to grasp that in conditions where hepcidin levels are high, the reduced export of iron into circulation drives iron deficiency and may result in anemia. So this occurs in a rare hereditary condition named iron refractory iron deficiency anemia, or IRIDA, where genetic mutations prevent the suppression of hepcidin. But far more common are the acquired anemias of inflammation that affect millions of patients worldwide with chronic inflammatory disorders. It's also important to note that anemias can also occur in a context of iron overload due to inappropriately low hepcidin levels. So in conditions of ineffective erythropoiesis, such as in beta thalassemia, the massive production of erythroid precursors signals the need for more iron in the system and therefore leads to a decrease of hepcidin levels, which contributes to systemic iron overload. So let's see how iron dysregulation may be tackled in these conditions. At the molecular level, hepcidin is mainly regulated by the BMP SMART signaling pathway, and it depends on communication between liver sinusoidal endothelial cells, or LSECs, and hepatocytes. So under normal conditions, BMPs are secreted by LSECs, and they bind their BMP receptor, inducing SMART phosphorylation and activation and promoting hepcidin expression in hepatocytes. The BMP receptors are associated with the co-receptor hemojuvelin, which facilitates the formation of the active receptor complex, therefore enhancing the downstream signaling cascade. This pathway has a physiologic break that becomes active under conditions of increased erythropoietic demand. So this suppressor is called matriptase 2, and it cleaves hemojuvelin, resulting in the formation of less or fewer receptor complexes, decreasing SMAD activation and also hepcidin transcription. When this break fails, due to mutations in the gene that codes for matriptase 2, hemojuvelin remains intact on the hepatocyte surface, BMP signaling proceeds unchecked, and hepcidin is always elevated. So this is the molecular basis of IRIDA, where the patients are profoundly iron deficient, yet they cannot suppress hepcidin, making their anemia refractory to oral iron supplementation. So currently, there are no consensus guidelines for treating IRIDA, and management relies on intravenous iron, which frequently provides only partial benefits because the high hepcidin levels still block the iron release from macrophages. And so this highlights the need for therapies that actually target the underlying pathway. So multiple approaches are now in development targeting different nodes of this pathway. And first, BMP ligands can be targeted directly, thereby intercepting the signal before it ever reaches the hepatocytes. And this includes small molecule inhibitors, monoclonal antibodies, and also other biologics that block BMP activity. Second, targeting hemojuvelin, therefore mirroring the function of matriptase 2, is also a possibility, and it has been shown with monoclonal antibodies. Third, the BMP receptor itself can also be targeted with small molecule inhibitors, thereby blocking the downstream signaling cascade even when BMP is bind. So although most of these agents remain in early development, anti-BMP6 antibodies and also BMP receptor inhibitors have advanced to phase 1 trials, not really in patients with IRIDA, but actually in patients with anemia of inflammation. And so it has been shown that treatment with anti-BMP6 monoclonal antibodies reduce hepcidin levels and increase serum iron in patients with chronic kidney disease. However, in this context of anemia of inflammation, there is another primary mechanism that drives the upregulation of hepcidin. So during infections and inflammation, immune cells, they detect pathogen components and also danger signals, which leads to the release of pro-inflammatory cytokines from immune cells. And in this context, IL-6 is very important because hepatocytes express the IL-6 receptor on their cell surface. And so macrophage-derived IL-6 promotes the activation of the JAK-STAT signaling pathway in hepatocytes, resulting in the upregulation of hepcidin. And although this IL-6 STAT signal is widely considered the most important driver of hepcidin upregulation in the context of anemia of inflammation, there are also other players that contribute to this upregulation. For example, macrophage-derived IL-1 beta signaling through the IL-1 receptor in hepatocytes also contributes to the upregulation of hepcidin. And additional players and pathways help explain the multifactorial nature of anemia of inflammation and why targeting a single cytokine provides only partial benefit for most patients. So current options for treatment in anemia of inflammation are quite limited. So we have erythropoiesis-stimulating agents that are often ineffective because the underlying inflammation suppresses both erythropoiesis and iron availability. And intravenous iron may provide some benefit, but because it does not address the underlying hepcidin elevation, iron remains trapped in macrophages. So we need alternative therapies that target the underlying pathways driving hepcidin upregulation. So for the IL-6 STAT3 pathway, there is intervention at multiple levels, both at the ligand and receptor level with monoclonal antibodies, as well as JAK2 and STAT3 small molecule inhibitors. And similarly, for the IL-1 beta pathway, there are also monoclonal antibodies available that can neutralize IL-1 beta or block its receptor. And what's particularly exciting is that some of these agents are already approved for particular inflammatory conditions. So studies have shown that rheumatoid arthritis patients treated with anti-IL-6 receptor monoclonal antibodies display decreased hepcidin levels together with increased serum iron and hemoglobin levels. And in patients with clonal hematopoiesis, IL-1 beta monoclonal antibodies decreased hepcidin and also improved iron parameters and hemoglobin. A different approach is to mitigate directly the effects of hepcidin on ferroportin rather than targeting its regulatory pathways. So as I mentioned earlier, when hepcidin is released into circulation, it binds the iron exporter ferroportin and promotes its internalization and degradation. And so this results in decreased iron export and the accumulation of iron within iron exporting cells. So to block the effect of hepcidin on ferroportin, there are currently 3 strategies. So one strategy is to neutralize hepcidin using monoclonal antibodies and other molecular platforms that bind and sequester hepcidin in circulation, preventing it from acting on ferroportin. A second strategy targets ferroportin directly using stabilizers that prevent hepcidin-induced ferroportin internalization and degradation, thereby keeping the transporter on the cell surface even when hepcidin is present. A third approach, still in early development, uses interaction blockers preventing the interaction between hepcidin and ferroportin. So most of these agents remain in early development, but initial validation data from studies in patients with anemia of inflammation shows promise. Most notably, a ferroportin stabilizing antibody not only improved iron levels, but also improved red blood cell parameters in patients with chronic kidney disease, therefore suggesting meaningful effects on erythropoiesis. However, the therapeutic strategies that I've shown you so far, they only target specific nodes of the hepcidin ferroportin axis. So in this sense, HIF prolyl hydroxylase inhibitors, or HIF-PHIs, they target multiple simultaneous pathways because they inhibit the enzymes responsible for HIF degradation, therefore allowing HIF to accumulate and activate the transcription of multiple target genes. So in the kidney, HIF stabilization increases EPO production, which drives the expansion of erythroid progenitors in the bone marrow. And these erythroid progenitors produce erythroferrone, or ERFI, an hormone that suppresses hepcidin transcription. So we'll discuss ERFI in more detail shortly, but for now, the key point is that HIF-PHIs indirectly reduce hepcidin transcription through this EPO and ERFI feedback loop. On the other hand, in the duodenum, HIF also upregulates the genes involved in iron absorption, thereby decreasing iron intake from the diet. Furthermore, this ERFI-induced suppression of hepcidin also contributes to increased dietary iron intake and also iron release from macrophages. So these agents are already in clinical use for anemia of chronic kidney disease, where they've demonstrated increases in hemoglobin and also reduced need for transfusion. They are now being investigated in cancer-associated anemia and also low-risk MDS, with early results showing an improvement in hemoglobin levels. And so this kind of covers high hepcidin anemias. And in the other side of the spectrum, in anemias with low hepcidin levels, iron regulatory pathways can also be modulated to improve patient outcome. So in the conditions in this category, the bone marrow produces massive numbers of erythroid precursors that cannot complete maturation. So the body senses anemia and responds by increasing EPO production from the kidney. This EPO production drives the bone marrow to expand erythropoiesis even further, attempting to compensate for the ineffective production. These expanded erythroid precursors, they produce enormous quantities of ERFI that is secreted into circulation and that suppress hepcidin levels. So ERFI works by binding to BMPs and particularly BMP6. And so when BMPs are trapped by ERFI, hepatocytes cannot activate SMAD signaling efficiently, and this leads to a profound reduction in hepcidin levels. And because of this hepcidin reduction, dietary iron absorption proceeds, and macrophages continuously export iron, and so iron accumulates and causes progressive organ damage despite the patients being anemic. So current treatment for these patients focuses primarily on managing the consequences. So iron chelation partially resolves the iron accumulation, but not really the underlying cause, which means that we are in urgent need for therapies that either restore hepcidin to control iron levels or address the ineffective erythropoiesis itself. So one of the therapies in development is related to matriptase 2, because the loss of matriptase 2 function causes high hepcidin levels, as we have seen in IRIDA patients. Therefore, blocking matriptase 2 in patients with low hepcidins as the potential to enhance BMP SMAD signaling and increase hepcidin production. RNA-based therapeutics and antibodies are being used to knock down and block matriptase 2, respectively. And so in consequence, hemojuvelin remains on the cell surface, and although ERFI can still sequester BMPs, whatever BMPs reach the receptor lead to a boosted BMP SMAD signaling cascade. And so hepcidin production, in fact, increases. These agents are in preclinical and early phase 1 and 2 development, with proof of concept studies in healthy subjects showing increased hepcidin along with decreased serum iron levels and reduced reticulocyte counts. There are also alternative approaches that bypass hepcidin production and directly target ferroportin. So the first uses synthetic hepcidin equivalents that mimic the natural hormone's action. So they bind ferroportin in macrophages and enterocytes, they trigger the internalization and degradation, and they block iron export. The second approach uses ferroportin inhibitors that directly block iron export even when the protein remains on the cell surface. So these strategies also remain in early development, with studies in healthy subjects showing similar effects to what we have seen with the inhibition of matriptase 2. So increased serum hepcidin with reduced serum iron levels and reduced reticulocyte counts. And so although these iron control strategies aim to manage iron overload and organ damage, they don't really address the root cause of the problem. And in this sense, so treatment with activin receptor ligand traps represents a fundamentally different strategy. So they bind and neutralize specific members of the TGF-beta superfamily that would normally activate SMAD2 and 3 signaling in erythroid progenitors that, in the context of beta thalassemia and related disorders, they drive the expression of genes that are involved in the inhibition of terminal erythroid differentiation. So in clinical trials, they've demonstrated enhanced late-stage erythroid differentiation together with meaningful increases in hemoglobin and significant reductions in the transfusion burden. However, the lack of more detailed mechanistic knowledge means that several questions remain. So in particular, how do these agents affect the activity of these ligands in other tissues? And most importantly, from an iron regulation perspective, how do they affect the ERFI-hepcidin axis and iron homeostasis? And so this brings me to my closing thoughts. I would like to remind you that we've talked about anemias that are driven by hepcidin upregulation and also anemias that have low hepcidin levels due to ineffective erythropoiesis. And what really emerges is a clear principle, is that context matters. So the same molecular targets may be used on opposite interventions depending on the underlying pathology. So what decades of basic research in iron biology gave us was the knowledge to have multiple nodes along these regulatory pathways that can be targeted therapeutically. And so this means that molecular understanding has directly enabled rational design of drugs. But obviously, important questions remain. So, for example, would combination therapies provide synergistic benefit in patients? What are the long-term consequences of chronically modulating these pathways? Can we develop biomarkers to predict which patients will respond to which interventions? And are there novel regulatory nodes we haven't identified yet? I don't have the answers to these questions, obviously, but what is clear is that we are moving from managing consequences to mechanism-based precision therapy, which I think will be key to change the lives of the people affected by these conditions. So I would like to end up by acknowledging my former group at the University of Heidelberg that taught me so much over the last decade. And also, I'm excited to share that I will soon start a new position at the University of Madeira in Portugal, where I'll continue my work in iron homeostasis, developing nanotherapeutics for anemia of inflammation. Thank you for your attention.
Please. Is there Andrea?
Thank you, Professor Marcus. I recall that the discussion of the full presentation will be at the end of this session. And so now we can proceed to the fourth speaker, who is the last speaker. And so we can start with a pre-recorded video. And the fourth speaker is Professor Mirko Pinotti. So we can start with this video.
So good morning to everyone, and I apologize for not being able to present directly this topic. And I thanks a lot Professor Maggio for the invitation, and it's a pleasure to be part of this intriguing initiative. So the topic is gene editing approach for the cure of hemophilia. And as you know, in 2022 and 2023, the FDA and EMA authorized 2 gene therapy for hemophilia A and hemophilia B. And this gene therapy, called gene augmentation, consists in the use of adeno-associated viral vector equipped with a normal copy of factor 8 or factor 9. And this virus can be injected directly into the venom, and it goes to the liver when it delivers the transgene, therapeutic transgene, and the hepatocyte can produce factor 8 or factor 9, which give rise to therapeutic levels into the bloodstream. While these approaches led to very promising results, there are still some limitations that consist in the durability, particularly for hemophilia A, because we see that over years there is a decline in the expression of factor 8, and also vector dilution, because the adeno-associated viral vectors are in the episomal state mainly, and they are not replicative. So if the liver proliferates and then there is a loss of the vector, it's called the effect of dilution. And for this reason, the gene therapy so far authorized are applied only to adults and not young patients. So one solution could be gene editing. So if you modified the gene and then this permanent change can be transmitted to the daughter cells. What is the rationale behind genome editing? The rationale is not new. It's just to learn from the mechanism, the basic mechanism of DNA repair. We know that when you have a double-strand break due, for instance, to a physics challenge, and then you can copy and you see the chromosome which is broken in gray and the chromosome in blue, which is the correct one. And then during the homologous recombination, there is an exchange of information and there is a repair of the broken chromosome. So you can copy from one side and transmit the information on the other one. The trigger is the double-strand break. So the rationale, the double-strand break. So we need the nucleases to target and cleave the DNA. And so over years, researchers have developed different kind of nucleases. I'm going to show you and talk about only the 2 main nucleases. The historical one, which are the zinc finger, is just a transcription factor that drives recognition of a DNA sequence fused to a nuclease called FOC. And the other one is the revolutionary, is the CRISPR-Cas9 system that led to the Nobel Prize to these 2 researchers. Well, if you do and you target with the nuclease the DNA and you perform a double-strand break, you have different alternative choices to manipulate the genetic information. If you cut and you don't give any template, and then you trigger the so-called non-homologous enjoining, and you are gonna insert or delete some bases, and you can use this system on the left to knock out a gene, to silence a gene. If you provide a template and you cut in the where there is the mutation, and then you can repair the gene and you have the gene correction. If you cut in another place, and I'm going to talk about later on, and you provide a donor, you can either provide a donor with homologous arm and you have homologous directed repair. But there is another technique called non-homologous integration that you can use a donor without the homologous arm. But don't care about this. So the rationale is that you need to cut in a precise way the DNA in a desired position, and you need a target nucleases like zinc fingers or CRISPR-Cas9, and you have to deliver, if you want a donor, and then you can have homologous directed repair in the desired position. Of course, you can repair the mutation, but you know, hemophilia A and B are diseases in which there is a very heterogeneous molecular pattern. So you would imagine, you should imagine so many donors to repair all different mutations, which is not feasible. What is more feasible is the target integration of the entire coding region to repair the gene or to integrate in another part. There is huge efforts from researchers in this field. I'm going to show you the most important result obtained so far. This is the pioneer study in Philadelphia in 2011, in which they exploited the zinc finger nuclease to cut inside the factor 9 locus, which is related to hemophilia B, and they tried to insert in the intron 1, sorry for some technical aspects, in the intron 1 the entire coding sequence of factor 9. They performed this experiment in mice, and as you can see, after the injection of the nuclease and the donor, there is a rise and an increase of factor 9 level that can be maintained over time. But look at the efficiency. Homologous recombination is not such efficient, and they obtain 4% levels of factor 9, which is good because it's not zero, you are increasing the levels, but it's not enough to have a really huge impact on the hemophilia phenotype. But what is important, what the proof of principle, is that if you compare the classical, the authorized gene therapy, in which there is a non-replicative vector, with the genome editing that I showed you, you can have this kind of outcome. If you take the mouse and you perform a partial hepatectomy, you force hepatocyte to proliferate. If you have a vector which is not replicative, the new hepatocyte loses the vector and you have a decline of the therapeutic effects. But if you modify the genome and then even if the hepatocyte replicates and then you are going to transmit the change to the others. And in fact, on the right, you can appreciate that after hepatectomy, there is no decline in levels and you persist. So you can treat neonates or infants with the genome editing, while with the other on the left is much more difficult. But the real revolution came with the advent of the CRISPR-Cas9 nuclease, which is a system much more versatile, in which a nuclease called Cas9 is driven to the target by a simple, very easy to design small guide RNA. So if you change the guide RNA, you drive the nuclease in the desired site very easily. And another very revolution, it was the discovery of the hyperactive factor 9 called factor 9 Padua, which has an increased specific activity, more or less 8 times higher than the normal factor 9. So even if you have the same level, the activity is 8 times higher. So they tried and they took factor 9 Padua and they exploited the new nuclease, doing the same experiment of before. But in this system, you can appreciate that instead of the 4%, they obtained 10% of circulating protein, but given the higher specific activity, the activity real was 100%, complete restoration of the coagulation. And this was obtained in neonates, mice, or in adults. It was um, um, not relevant whether it were adult or neonates. Good, but we can do better. And the real very fantastic change was, why don't we target and we integrate factor 9 instead of the factor 9 locus, which is a weak gene and a weak promoter, into the locus of the albumin. Albumin is very high expressed, is one of the most concentrated protein into the bloodstream, is a liver-specific expression, and if you lower a little the albumin level, are well tolerated. So they used the albumin locus as a safe harbor to integrate the therapeutic transgene, either factor 8 for hemophilia A or factor 9 for hemophilia B. And these are the results in both neonates or adults. And you can lower the amount of the AAV delivering the nuclease and the donor, and you have a really fantastic effect over time. However, yes, and the albumin expression was more or less maintained, so it was safe. But there is a concern in using the nuclease, because if you deliver the nuclease with a vector, like a viral vector, and then the Cas9, the nuclease, persists inside the cells, so the risk of cutting in other places, the so-called off-target effects, is there and can raise concerns. Can we be safer? The answer is yes, because you can use and deliver the donor, which is without promoter, is not expressing factor 9, but just the cassette with the AAV vector. But you can deliver, as for the vaccines with COVID, you can deliver the CRISPR-Cas9 system as mRNA with lipid nanoparticles. So the lipid nanoparticles can deliver the mRNA. The Cas9 is expressed inside the cells, but the mRNA is rapidly degraded, and so the Cas9 cannot persist, and the risk of target effects. And this approach, in the combination of lipid nanoparticles and AAV vector, is exploited now as produced really promising results in mice and non-human primates. And now there is a clinical trial investigating the efficacy and safety of this approach in human. Can we be even smarter? The answer is yes. You know that for hemophilia A and B, one of the approaches pursued currently is to downregulate the physiologic inhibitor antithrombin by a small RNA. And in this way, you downregulate the anticoagulant pathway, and you can push the coagulation. But as you know, antithrombin is a protein highly produced in liver in hepatocytes. So it could be, like albumin, another safe harbor. And in this way, researcher did the targeted insertion of the transgene, either factor 9 or factor 8, in the locus of antithrombin. In this way, you can have a double effect. You are going to raise the level of factor 9, in the case of hemophilia B, and you are going to downregulate antithrombin. So you push the coagulation on the left, and you push also the coagulation by reducing the anticoagulant pathway. And results, like shown here, with the double system, the double delivery system of lipid nanoparticles and viral vector, are really promising in hemophilia B and also in hemophilia A, in which you can see, on the right in blue, you have the higher survival rate after injury of mice treated with the gene editing as compared to the others. But the system that I showed you consisted in the direct gene editing. So you were injecting by intravenous injection the components of the editing. Can we be more safe and use cells from patients transported outside, modified, and re-injected? Yes, in this case, you speak about cell therapy, not in vivo gene therapy. And this is a very intriguing study in which researchers, instead of using a hepatocyte, took as a factory for factor 9 the B cells. So they took B cells from patients, they manipulate B cells for the target integration of the factor 9 Padua, the hyperactive version, into the CCR5 locus, which is highly expressed protein, into the B cells. And then they took these engineered cells and reintroduced in the patient to permit the homing to the bone marrow without preconditioning. And even this approach gave nice results in mice, and also this approach can permit also a redose of the modified cells if needed. And also in this case, there is a clinical trial entered in phase 1-2, and I'm really curious that I believe we are all curious to know the results in patients. So I showed you the genome editing based on double-strand break, but there are also systems entering into the in vivo studies that can be extremely accurate and without double-strand break, which is really sometimes risky. These two systems are called base editing, in which you can change one base to another and you can perform single nucleotide changes and you can revert point mutation. And also the other one is called prime editing, in which I don't want to enter into the details. It's very complicated, in which you can make different changes in the same region, and you can replace or even correct small deletion or small insertion. And you can correct in this way all type of point mutation or small mutation. We performed a study in hemophilia A with base editing, and I'm going to show you just the flavor. We were able to rescue 4 mutations. I'm going to show you a couple of mutations, but now we have preliminary data for much more mutation, either in factor 8 or factor 9. And as you can see, we performed this experiment in cells from patients engineered with the editors, and there is a dose-dependent increase of the secreted factor 8 in the presence of this stop mutation on the left, or in the presence of this misses mutation on the right. And the increase in protein level are paralleled by the activity, which is demonstrated by the decrease of coagulation time when you supplement deficient plasma with the protein produced by the engineered cells. Is not just an example mine, but with the base editing, let's see 1, 2, 3 example. With the base editing, there are clinical trials approaching different diseases, and here I'm just summarizing some of the most relevant at present. So I walk you through the genome editing. I would like just to thank all the agencies that have supported our research in the field, our collaboration, and really I thank you for your attention, and I apologize again for not being able to interact with you Saturday morning today, since you are listening to me. Many thanks and best wishes to all of you.
Thank you. Thank you, Professor Pinotti. We are perfectly on time, and I thank you all the speakers for that. Now we have 10 minutes for the discussion of the 4 presentations, and I invite everybody to ask their questions, and particularly the faculty who are connected, and we can start this discussion now. Any questions?
May I start with a question for Oriana and compliment for the talk. You address a very important problem, that is the use of combination treatment, and that is the future, and everybody I think is convinced are convinced that is the future, but the problem is who will be perform the trial, the clinical trial. We have to ask to wait for the company, or will be or it is necessary, that is my opinion, that scientific academy and opinion leader share to start some trial, some independent trial on this combination treatment, because now today we have a lot of this opportunity. I am thinking about luspatercept and josuria, for example, because the mechanisms are different, or other possibility are inhibitor of epsilon, or other drug that are used in hematological malignancy associated with other like luspatercept. So I would like to have your opinion about. And then there is a question by Dario Martino, Andrea, that you can address.
Yeah, so thank you for the question. I'm blocked. I actually I agree with you. I think we have somehow to push this further. Otherwise, what we see, and actually what we see on the iron biology field, is that most of these therapeutics are being tested in patients with anemia of inflammation, right? Chronic kidney disease, autoimmune diseases, because there is a lot of patients with these diseases. So the pressure goes into treating diseases where you have higher numbers of patients. And I think that the early results in patients with anemia of inflammation are very good with these agents. So this kind of pushes further to try combination therapies in patients, for example, with beta thalassemia, that otherwise, if we don't push it, we will not get there. Again, talking about combination therapies, for example, last year there were the papers from the group of Stefano Rivella and Antonella and I with the combination therapies with the activin receptor ligand traps and TMPRSS6 inhibition, or with the TFR2 inhibition and erythroid progenitors, and the results were very good, but it still is in preclinical assay. So I guess if we push this further, we'll make get more benefit into the patients.
Okay, thank you. My comment is that these talks were excellent, of course, and they showed that the progress in this field is extremely rapid. And this progress is, of course, an important advantage for the patient, for science in general. But on the other hand, I see that the regulatory agency do not keep the pace of the progress which is so rapid. And so this raises a problem because while we see that new treatments enter, become available and are being tested increasingly in so many patients, and the proofs of the benefits are well demonstrated. This is an important prerequisite, of course, but we have a problem not only in Italy but in Europe in general, that regulatory approvals are not so fast. And so I don't know how long it will take to the EMA or AIFA, which is the Italian agency, to evaluate these innovative treatment and then to make them available on the market and on the patients, to the patients especially. And this is a point, this is specific to gene therapies in general, because later on we will see that gene therapies are now quite numerous. We have more than a dozen of gene therapies that have been adequately tested in clinical trials. But the availability for the patients is delayed. And we must discuss this problem because its practical implications in the real world, of course, are particularly important. I have no solution for this problem, but I wanted to stress that this is an important problem.
I think that there is a question by Dario Martino from the audience. The question is, good morning everyone, and thank you for this occasion. Maybe you are already said, but my question is, after the socio-economic approach, what could be the impact directly for patient? The age and the clinical access for treatment could be extended, for example, to who are actually excluded to other approaches due to their morbidity. I don't know, Andrea, what would you like to comment on this? And my brief comment is that extension, it depends on the company that are involved in the regulator trial. For example, for gene editing, they decide to include only patients from 12 to 35 years. And I ask Franco Locatelli which was the rationale, and he stated that the rationale was to include a cohort of patient with a low chronogenicity and risk. So this means that this cohort could be extended, but it depends on the on the study that are that the company could perform. The other question different is if you use drug. If you use drug, the extension is more. I think is greater and may include the patient behind the 35 years. But the problem of the extension is due to the design of the trial sponsored by the companies. I don't know what do you think, Andrea?
Well, my comment is that in this context, our priority should be to encourage the conduct of clinical trials, the participation in clinical trials, and the development of the research that we have seen before. And we must admit that all clinical trials in this area are industry sponsored, and there are virtually no exceptions to this rule. In my opinion, there should be also some promotion of independent trials by national institutions, and some investment should be given also by regulatory agency, by the Ministry of Health, because at present time all research is being conducted and is on the shoulders of the companies alone, and there are very different methods for encouraging new trials, and one method could be the direct sponsoring and the investment by national agencies in this area. And the other method could be to be quite generous in recognizing the price to be given to gene therapies, because the price that we recognize to the patients, to the treatment of the patients, must include the research on that agent, which is an essential component of the development of this area. At present, I am afraid that the investment are all of private origin and are all made by the companies involved in this in the production of these treatments.
Okay. If there are no other questions, we can move to the other session. I don't know. And the next chair is Paolo Foggi from Haifa. Is connected?
Yes, I am.
Okay.
So
Thank you, Paolo.
good morning, everyone. So I'm happy to introduce this second session, which is more about also the involvement of regulators and how regulators can improve the access. So I will give the floor to Antonella Isgro, which is an hematologist and a colleague who works in the agency. It's also representative in the Hematology Working Party at the EMA, so she's involved also at the EMA level. and she will have a talk about the challenges and opportunities in drug development for rare disease from the regulatory perspective. So, Antonella, the floor is yours.
Thank you. Do you see my screen?
Yes, perfectly.
Okay, thank you for the invitation and opportunity to speak at this meeting. I cannot move on this. Okay, this is my disclosure. Rare disease represent a unique challenge for drug development because of their low prevalence and complex nature. The Orphan Drug Act in US and similar legislation in EU introduce incentives such as market exclusivity and financial support, which has been critical in stimulating innovation. However, despite this measure, only a small fraction of rare disease, about 5% of more 8,000 rare disease, have approved treatment. Regulators are working to address the gap by promoting early dialogue with the developers, streamlining process, and accelerating approval while maintaining a robust evidence standard. But global collaboration remains essential to meet this unmet medical need. Early engagement between regulators and sponsors is key to identify potential issues and accelerating development. International collaboration such as Access Consortium and Project Orbis help harmonize regulatory approach and share information. We also involve academia, no-profit organization, small pharmaceutical companies and patients to ensure that decisions are both scientifically sound and patient-centered. This is the legal framework to incentivize the rare disease drug development. Orphan drug legislation provides a framework to encourage investment in rare disease treatment. This law defines criteria for orphan designation, offer market exclusivity for a specific period, for example, 7 years in US, 10 years in EU, but this specific point is under revision with the new pharma legislation, and provide financial incentives such as tax-free credit and fee reduction. Together, these measures reduce financial risk and make rare disease drug development more attractive to industry. In particular, orphan drug designation is a critical mechanism for rare disease drug development. Orphan designation provide additional benefits such as fee reduction and protocol assistance, and these incentives are particularly important for academic institutions and smaller development. Accelerated regulatory pathways such as priority review and conditional approval are also important for bridging therapy to patients faster. These mechanisms allow flexibility in accepting, for example, surrogate endpoint and interim data, provided that post-approval commitments are in place to confirm clinical benefit. Italy has implemented several mechanisms for early access to medicine, including specific law. As a result, Italy ranks among the highest in Europe for orphan drug availability. However, as discussed this morning, high price strategies pose a challenge to a sustainability of a healthcare system, making necessary to strengthen health technology assessment and explore innovative price models. But other speakers will be involved on this issue today. The other problem and the first challenge is in relation to the current clinical trial for rare hematological disease. Clinical trial for rare hematological disease face significant challenges. Small and heterogeneous patients population make traditional randomized control trial difficult. Recruitment barriers such as socioeconomic and geographic factors further complicate participation of these patients. Sickle cell disease is a clear example of a systematic underfunding and limited therapeutic success despite its global prevalence. We urgently need adaptive and inclusive trial design to address these complexities. However, hemoglobinopathy are also an opportunity for academia. In this context, hemoglobinopathy could be considered an example. Over the past few years, there has been an increase in understanding of the sickle cell disease pathophysiology, with several treatment approaches proposed for modulation of hemoglobin polymerization, prevention of vaso-occlusion by inhibition cell-cell interaction, prevention of endothelial dysfunction, and modulation of inflammation. The same for thalassemia, addressing ineffective erythropoiesis and hemolysis, modifying iron metabolism, or altering globin gene. There is a vital need to design and conduct clinical trials for new drugs or drug combination to optimize the long-term management of these patients, and clinical trials are the main source of evidence to assess the benefit-risk profile of medical products in marketing authorization procedures, but there are some limits. Hemoglobinopathy are rare and phenotypically diverse. A trial that targets a clinical problem occurring in a subgroup restrictive of eligible participants, and the investigators may overestimate these clinical trials so that a feasibility problem could exist in clinical trial in rare disease, or the clinical problem addressed by specific trials such as VOC or RCS may be an acute event in sickle cell disease with a frequency that is not predictable. But the evidence from a regulatory perspective, from academic and pharma industry research for translation need to be solid in every step of drug development until approval and patients access. The first challenge could be related to the trial population and design. For regulatory perspective, the participant enrolled in the research study should reflect the population affected by the condition or those for whom the treatment or intervention is intended. Hemoglobinopathy have a substantial genotypic and phenotypic heterogeneity. Moreover, the variability in sickle cell or thalassemia expression is only partially explained by genotypic, as socioeconomic and environment factors can also contribute to this heterogeneity. The representativeness of early clinical data as well as an impact on pharmacokinetic, pharmacodynamic, efficacy and safety data based on genetic variability should be taken in account. About the clinical trial, randomized clinical trial is still the gold standard, but in some cases, randomized clinical trials are unfeasible or unethical. In these cases, a single-arm trial may be acceptable, but they should be strong and self-standing. An external control arm in support might mitigate the uncertainties. External control arms has been accepted by the FDA in many rare disease approval, and EMA also consider them under specific condition. Success depends on methodological rigor, transparency, and early engagement with stakeholders. Sponsors should carefully access data sources, define protocol clearly, and justify statistical method to ensure regulatory acceptance. On this specific issue, EMA is currently involved on a reflection paper on the use of external control arms. But In general, external control arms are important tool in rare disease drug development when randomized clinical trials are not feasible, but these studies, these pragmatic trials could provide fit for purpose comparator data supporting regulatory decision in rare disease, but proactive engagement with the regulators is essential to address methodological challenge and ensure successful outcome. The second point related challenge in rare disease is related to the endpoint. Regarding the efficacy primary endpoint, it's important that the selected clinical outcome assessment are fit for regulatory use, that they are valid assessment of important and relevant aspect of the disease, and that the input is obtained from clinicians with expertise in caring for patients with the target disease. When no validated clinical endpoint or biomarkers are used, they should be validated before being accepted in a clinical trial. In the era of gene therapy and gene editing, transfusion independence and on the hemoglobin increase have been considered validated surrogate endpoint as a clinical benefit in terms of reduction of morbidity and mortality has been demonstrated in this specific population. Some problems there are about the endpoint in sickle cell disease because the vast majority of drug trial in sickle cell patients have used acute pain episode of vaso-occlusion crisis as the primary clinical endpoint. This is not surprising as acute pain episodes represent the most frequent reason for medical contact by patients with sickle cell disease. But VOC are the main causes for the low success rate of sickle cell disease clinical trial, given the complex pathogenesis of this complication, as well as the difficulty in accurately quantifying this episode. Most sickle cell patients, more than 94%, have infrequent VOCs requiring hospitalization, and they are excluded from VOC-focused trial. Moreover, sickle cell patients can have lifelong complications in addition to the pain, for example, anemia, acute chest syndrome, stroke, pulmonary hypertension, so that we need specific new endpoint for sickle cell disease. The process of EMA, marketing authorization, is a complex process with specific timeline, in which regulatory decisions are based on absolute benefit risk assessment, considering the comprehensiveness of efficacy and safety data and the uncertainties on both sides. In rare disease, pre-approval data may be limited, so post-marketing studies and registries are essential to collect additional evidence, but discussion on real-world evidence should start early with regulators. In this context, real-world data and registry play a critical role in rare disease research. They help improve diversity, enable long-term follow-up, and support regulatory decision. In this context, Darwin is an international platform related to the use of real-world data to generate real-world evidence, aimed to generate real-world evidence to inform EMA and national regulators. And this is very important also for to collect evidence in post-approval phase. EMA offers several tools to support innovation, not only limited to the orphan designation, but including innovation task force, the prime scheme, the scientific advice. In particular, the scientific advice can help during all the phase of drug development with scientific advice on specific product, protocol assistance, qualification opinion, qualification advice, and priority medicine scheme, which offer in particular support for the development of promising medicine that target an unmet medical need, but after Paolo Foggi speak about on this. This represents a multidisciplinary platform for initial dialogue on innovative methods, technologies, and medicine, including the identification of biomarkers, patients report outcome with the involvement of the patients, surrogate endpoint, but with high standard of evidence in the interest of the patients and of society with the cost. IFA is involved in many assessment activity of KIMA, also with the participation in specific committee and the working party. Here you can see a series of national EU activity of IFA in support of innovation, including national scientific advice, EMA scientific advice, the involvement in European Innovation Network and its group, network data sharing group, and specific web page in collaboration with the EMA dedicated to academia. IFA and EMA promote early dialogue and innovation support. Bridging the gap between academia and regulator is essential, as well as sharing data and involving patients to ensure that rare disease treatment are developed in and meet real-world needs. In conclusion, medicine for rare disease is a global priority. While regulatory incentive and flexibility approach have driven process, challenges remain in access and pricing. But adaptive regulatory strategy, sharing the data to generate common action at the European level, and collaboration among all stakeholders are key to ensure optimal care of these patients. Thank you.
Many thanks, Antonella. Very clear as usual. I think that we have time for discussion at the end of all the presentations. So in the interest of time, I will proceed. And indeed, in the program, it is planned to have my presentation. So I'm going now to share my screen. I hope you can see now. And so I think that as Antonella was alluding to, of course, it is very important that regulators and developers interact. We are well aware as regulators, and Andrea before put, so to say, the regulators on the spot on the fact that perhaps we are the bad ones who delays the access. I would not say so. Of course, there are challenges, and we are trying, believe me, to do our best not to get it happen. And I will try to explain the next few minutes how. We are well aware that the road to a drug to a right to patients is full of challenges, and this is not. As you can see here, there are a lot of problems that go from scientific, financial, and also organization and technology transfer is one thing on which I guess there is much room for improvement. Stakeholder coordination, of course, adapting the regulatory framework is important, and then, of course, there is the problem of the market access and reimbursement, which is, of course, a barrier after the regulatory approval. I think that what we are trying to do is that from our side, it's pretty clear that regulators must adapt to the technological advance, and we must not be a block. we never should be the block. That means that we need to be in contact with. But on the other hand, it's also true that developers should understand that they need to adhere to regulatory standards from the early stage of development, because there are too many instances in which something is delayed because it's not done properly since the beginning. And this is mainly from academic researchers that start, and of course that's really the need for having a mutual exchange, for us to adapt the regulatory standards to keep the pace with innovation, always trying to ensure a proportionate risk mitigation measure. We as regulators, we not to ask for the impossible. On the other hand, we need to to draw a line where the regulatory acceptance is enough. And I guess that in this dialogue, we need together identify the most critical intervention and prioritize them accordingly. Important to say that regulators in EU works together. There are a lot of national competent authorities in EU in each member states, but just to ensure that we try to work together under the umbrella of a EMA. And so I guess that this is happening in a quite effective way. And this is just to make you aware, in case you don't know, this is the network of regulatory agencies strategy to 2028. And you can see here that the strategic focus areas, and you can see here I just want to point out to you more on the accessibility. Of course, it is important, and this should be looked more in view also of the new HTA regulation, and the HTA regulation also calls for a higher engagement from the very early stage between stakeholders, regulatory agencies, and HTA bodies. And this is expected to, in a way, to speed the access, to ease the access to drugs, since issues can be discussed and probably solved at the earlier stage that compared to what's happening right now. And the other point that I want to highlight, and more on the focus of my presentation, because it's what I'm currently doing in the agencies around the regulatory science, innovation, and competitiveness. And this is the kind of work that is tried from the regulatory side to accelerate the translation of innovation, and also make you more attractive for researchers and for industry also to invest here, and also more friendly for researchers to develop new therapeutic approaches in you. When it comes to the agency, we'll try to translate this vision in 4 strategic pillars that are all interlinked together, if you wish. So you do not see these pillars like silos, but something that is really interlinked. Important to provide support tools for developers, and I will try to explain a little bit to make you aware of what is available for you to interact with the agency. So there are innovation meetings, scientific advice. So to have an early dialogue during the development of a new technology or a new product, so that we can start an early discussion, as Antonella was saying before. Then there is the pillar of training and capacity building. And this is really for us to provide regulatory training for researchers at universities and give our contribution to try to enhance the translational impact of academic research. But on the other side, I think that we need to get also training from academia on emerging innovations in order to keep the pace of innovation, and also to have the expertise and knowledge also in our environment. Sorry, I did something that I never see do not see my presentation. Can I check whether you see my presentation?
I can see the presentation.
Okay, sorry. Okay, it disappeared to me. Now it's back. Okay, sorry. And the other thing is regulatory science. So engaging with stakeholders to identify innovation and address regulatory gaps and adapt standards for implementation. And then, of course, it is important the international collaboration, because it is of utmost important that we try to harmonize what we do here and what we do in EU, also with the global regulatory landscape. Regulatory science. The model is what we called internally the IFA lab. Lab is listen, analyze, and build. So being in a listening mode is important to understand what the upcoming innovation and what are the perceived barriers and proposed solution. And then the analysis is to see, okay, what are the regulatory gaps? What we should do to improve? What are the priorities from our stakeholders, and what are the actions we need to do to improve our capacity in terms of expertise, in terms of infrastructures, and try to have an input in risk assessment, and to understand when we plan to make some changes, what are the implications. And of course, on top of this, then build, so recruit, renew digital infrastructures, update our guidelines and policies, and adapt frameworks. And the aim is to always get the best trade-off between accelerated access and maintaining high quality and efficacy safety standards, because it is in our remit to protect the health of the people. So, just to give you an example, what is to be in the listening mode? Of course, this is happening in many ways, but one is the experience of the horizon scanning reports, which is done by regulators, but also involving scientific experts in the field. These are some examples of what has been done in the context of EU Innovation Network, fecal microbiota transplantation, genome editing, nanomedicine, healthy aging, companion diagnostic. There is something new ongoing. So I think that this is very important, because this allowed us to understand what's coming and try to analyze what the gaps are and how we can improve in this regard. In terms of training and capacity building, I want to share with you just some experience that we have, especially in the being involved in European projects and the stars. It was right. It was a project that ended, but it was a project trying to promote regulatory awareness and knowledge to academia, and also now we are involved in the IncreaseNet project. And the IncreaseNet project is aimed to support increased capacity and competencies in the EU medicine regulatory network. And we at IFA are the leading of task 1, which is indeed fostering collaboration with academia. And you know that in rare disease and hematology rare disease, it's very important, the contribution of academia. And I guess that what we are trying to do is try to develop a model and to standardize a model in which across Europe academia could provide on a regular basis training on innovative topics to regulators, because it is very important that we are so humble that to make us learning from the academia. And we have a call for expression of interest, and this and selection is being completed, and two topics were selected based on the relevance, and when on RNA therapeutics and organ on chip. So just to say that this is the first experience in which we try to build something in which we learn from academia. Then support tools for developers, and already Antonella said something about there are a lot of possible interactions that could take place across the medicine life cycles, and there are a lot of tools that are made available both at the EMA level and the IFA level and the National Competent Authority level. So this is just to give you a flavor, and I will try to expand on some of those, but just to I think that this is to just to make you aware of how many kind of interactions are possible, and I invite you really to have a look to the EMA website and the IFA website, because there are a lot of information important that you could look at and relevant. And so this is about the main that I will touch upon, the Innovation Task Force and the SME Office, academia support, and also just to remind you also the Prime scheme that was mentioned before. And when a drug is promising, it can have an early interaction, an early appointed rapporteur at the CHMP, and strengthen the interaction. And this is something that could then end in an accelerated approval. I will not expand more on that, but just to make you aware, this is an important also part. With regard to the innovation meetings, this is available both at EMA level or at the national level, and it is a very early informal dialogue which can it's free of charge, so you can ask and access to this kind of interaction. This is mainly dedicated to very early, when a new technology is being developed, to just taste the water with regulators, understand whether which are the appropriate guidelines to take into account, or whether guidelines are missing, where to go to be on track during the development, so that then it can be translated, really your research can be translated in something that could be acceptable from the regulatory perspective. So I guess it is what most important is this kind of early dialogue is happening, and over the years we had many interesting interactions that were very important. They were important for developers to be on track and to align the development they were doing in order to be compliant with regulatory standards, and also for us to get the feeling of what was new, and perhaps some guidelines were missing, and something need to be adapted in order for the regulatory systems to be ready to accept this. So I guess this is a really brainstorming style meeting, and I guess that's really important, and I really encourage you to consider this kind of interactions. Then the scientific advice. The scientific advice is a little bit more, so to say, formal. Formal, I mean the fact that when you get in the scientific advice, then in future interactions with regulator, you always need to refer to this kind of interaction and provide the relevant documents and the scientific advice that you had. And this is important to know, and this is also more focused on specific development. So while the innovation meeting might be more general on the technology, even also on a specific development, this is more usually dealing with a specific development and could deal also with the quality aspects, non-clinical aspects, or the clinical aspects, what would be the patient population, the proper comparator, if not possible to have a randomized trial, what is the proposal, what is the endpoint. So in a way, trying to get an alignment between what is the proposal of the developers and what could be something that the regulators could see as a proper way forward. And then, as of course, is always trying to find a trade-off from which it is feasible and what could provide the highest regulatory evidence to make in the end the benefit risk decision at the time of approval. This is just to make you aware that this can happen at the national level or a European level. There is also this Act U Priority Action 7 is looking at accelerating clinical trials in DU. And one important thing that we introduced is the fact that it is possible now to seek a scientific advice, not just to check whether the development is fine for the marketing authorization application, but also if it is fine for the clinical trial approval, which is something new in the European landscape. Usually, if you go to FDA and discuss with the FDA, you discuss with the regulators, and then they say, okay, this is fine for conducting the clinical trial, and it's also fine what you're doing to support a marketing authorization application. Currently, this was not the case in Europe, so it's something new that we introduce. We have the simultaneous national scientific advice for clinical trials that are being conducted in more than one member state. You could come or centrally to the EMA, to the Scientific Advice Working Party, or you could target 3 or 4 member states only, especially if you're not that interested in discussing the marketing authorization, or rather the clinical trial application. And so you could get simultaneously a feedback on the adequacy from the regulatory perspective of what you're doing simultaneously from 3 or 4 member states in which the clinical trial is going to be conducted. So I guess it's enlarging the landscape and the tools that are available. And it's not in this slide, sorry for that, but I also want to say there are also the joint scientific consultation. This is part of the HTA regulation. In this case, you can have both the feedback from the EMA at the Scientific Advice Working Party level and the HTA bodies about the adequacy. So in this case, you might discuss whether the clinical development that is being done is adequate to support both the regulatory requirements and the HTA requirements. And this is a part of the HTA regulation that I was referring to before, and is expected to improve probably and accelerate the access to drug in the end. Qualification opinion, I just want to shortly mention, it is important, especially when one is developing a new endpoint or a new tool for enrichment of the population, a new prognostic or a new biomarker or a new technology applied. For instance, it could be something based on artificial intelligence, or also if you want to qualify a registry for a given context of use to support the conduction of post-marketing studies, so to generate evidence from regulatory purpose, there is the tool support tool of interaction that's about the qualification of novel methodologies. Anyone could apply, and there is also here a qualification advice, and it could then result in a qualification opinion. A qualification opinion means that this new tool, this new biomarker, this registry for a given context of use, this artificially based technology or whatever is qualified to be used in a development for the given context of use. So this is something that does not relate to a single development, but something that could be applied more broadly. And there are many examples of that, and I invite you to have a look on the EMA site. So there are many of those. Then I wanted just to highlight to you this tool of the academia briefing meetings, which is, I think, is very important tool that is available from academia to interact with regulators at the central level, at the EMA level. And also it has been launched the European platform from regulatory science research, which is a forum in which regulators and developers and the researchers can really discuss and try to shape the future of the regulatory approaches in innovative fields. So I'm going to the conclusion of my presentation. What we aim is to guarantee timely access to innovation for patients and provide regulatory support tools from the early stage of development. We try to work to facilitate innovation while on the other hand ensuring compliance with regulatory requirements. To do so, we need to keep up with the pace of innovation, evolving the way we work probably, and promote regulatory science. And this can only done honestly with the collaboration between regulators and developers, because it is essential. And we have shared goals clearly. And I was listening to the presentation also in the previous section of this interesting meeting. I think it's pretty clear that we cannot proceed without mutually listening and working together for the common good. So this ends my presentation, and I thank you. And so I think that I stop sharing, because being a panelist and the chair, then I think that I need to give the floor to the next speaker, which, let me check, I think it's Armando Magrelli. And of course, is another IFA colleagues. This has been at the comp for many years, so he's really an important player when it comes to rare diseases. Has been the vice chair also there, and now is in the agency. So it's my pleasure to introduce him. We'll talk about the approaches from artificial intelligence. Armando, please, the floor is yours.
Thank you, Paolo. Thank you for this invitation. I want to thank Professor Maggio and also all the network. Actually, I'm very happy to be here. I try to share my screen. It should be this. Just for checking if I'm in the right page. I hope that you can see the slide.
Yes.
Okay, great. So I have been asked to explore and to see what actually European Medicine Agency, other agency, and actually worldwide. I will not cover worldwide the approaches, but I try to give you a rapid overview of what's happening regarding the AI and how regulators, to some extent, try to address this new field, because as mentioned by Paolo and also by other colleagues, AI is going to change our way of assessing, to some extent, new medicinal product, but also is changing how research will be conducted, and also how this information, these tools should generate solid and robust evidence. This is the title that actually I tried to modify when I was preparing, but it's just to tell you that the field is growing and many. Let me check. Yes, this is my declaration of interest, and we can start from what regulators do with regards the AI. As you may know, regulators, agencies around the globe are moving quite faster when they recognize that actually AI is changing the current panorama. And actually, just to highlight you that if we compare EMA, the European situation with the FDA, there are similar approaches, but to some extent are a bit different because obviously the scenario is different in terms of countries and so in terms of continents. So we can start from Europe, in which we start to approach the big data with the DARWIN project in the past years, and then the data AI program, which started in 23 along to 2028, and then generation of some ethical and technical guidelines. While in the US, there was an approach in accelerating the assessment of AI technologies. Also, there was a movement with regards to the transparency of the algorithms. We will see later on. And also, they state very clearly that the human supervision should be mandatory in all AI technologies. These are some of the act and the paper and documents which were generated along the years. This is currently the EMA and HMA Network Data Steering Group, which provide a data plan, a work plan for the next 3 years. And then you can see the reflection paper on intelligence, on the artificial intelligence, starting in 2023, but actually published last year's. And we also have on the other side of the ocean, we have the consideration for the use of artificial intelligence to support regulatory decision, and also how the artificial intelligence relate to medicinal products and the FDA. Among these, we have, as mentioned by Paolo just a few minutes ago, we have the qualification of AI technologies, which recently this year, early this year, the agency established and qualified, given a qualification opinion for an artificial intelligence-based system with regards non-alcoholic steatohepatitis. But this is just to tell you that actually there is a lot of movement, and these are obviously qualify the methods which can be used and agree in the process of assessing medicinal products. And finally, well, we will touch just briefly, but just to tell you that despite the let's say the central approach to the AI, also single agency are developing their own AI-based tool for accelerating some aspect of their work. This is from the Swedish Medical Agency, but we also have from Finland, the other country, which are AI tools based on usually on LLM language, try to, let's say, sort it out a large number of documents and analyze a large number of documents for, let's say, giving some information for the users. Oh, sorry, I got too fast. Why it matters? Why AI matters? Although it's out of that it matters. What is not out of that is the how we use AI. So for them, from the hematological point of view, you know better than me, but we can use in blood smear analysis. We can connect MRT with the flow NGS pipeline. We can use AI for interpreting NGS data in ML, ILL, and many other disease. More advanced is the use of AI in PEC and also in MRI. In lymphoma, we have many examples. And obviously, EMA is reviewing all these AI tools for approval. This is briefly the EMA's roadmap for the AI use. As I mentioned before at the beginning, there is the EMA Strategy 2028, which is focused on integrating AI mainly in trial and in safety monitoring, and also try to harmonize all the framework within Europe. We have many cases in which these are particularly important, such as using in trial for including specific marker or for having image-based endpoints, and also safety monitoring, for example, in CAR-T treatment, in which AI has been demonstrated, not yet validated, but has been demonstrated very useful in depicting patterning for ICANS and CRS. What is the main point? What we have to keep in mind? EMA, but not only EMA, insists on transparency of the algorithm in the AI and the validation, which is always under the human oversight, and which actually, as mentioned before, is directly linked to the EU AI Act to ensure consistency and safety. But we will see in a while some tiny example. So as I mentioned in the first slides, we have the reflection paper on the use of intelligence or artificial intelligence, which cover all the life cycle of the medicinal product. Actually is a very broad paper, but actually establishes some I wouldn't call guideline, but some principle which depict the next framework. Obviously, we have many implications in the hematology field, but also in many others, in which the AI-based, for example, in MRD, in marrow imaging and also in risk model can be applied, they can be used. For example, I found an example of the MDS risk model using AI, which has been recently published. But this is just to tell you that actually there is a huge movement, and maybe some of the next speaker will touch upon this specific example, a specific experience. But just to tell you that European Medicine Agency are taking, let's say, having let's say, a strict control on what is happening around with regard to the use of AI. So when we look at the use of AI in, let's say, the old AI in drug discovery and non-clinical, we have many experience. We have a well-established, let's say, use of AI, especially in drug discovery, in sorting out toxicity, interaction. There are many examples, and actually are quite, let's say, well established from a scientific point of view. So, in this respect, when we look at drug discovery, where we look at drug definition, toxicity profile, or modeling, or new drugs, or whatever, the scientific knowledge is quite advanced. That actually, it's. I mean, this is reality, and there is no major, let's say, issue apart the fact some concern that we would see at the end of my presentation. One of the aspects, as I mentioned before, is how we can make a profitable use of the AI, for example, in a clinical trial. There is an issue with regard an issue. There is a possibility to use the data-driven recruitment, as mentioned before, in which the AI can mine or can dig around the health electronic medical records to find patients with specific mutation, for example, or other specific features. To some extent, it's also possible to improve, apart of the speed of enrollment, also to foresee how the trial will go, have some kind of prevision, and also try to establish an equity in terms of representation of the outcomes. For example, there is this approach for finding TP53 mutant patients from databases, which to some extent boost the possibility of enrollment, and also AI is quite useful in terms of, for example, analyzing multiple centers for reaching the numbers of patients, also for having let's say a timeline of accrual of the patients. With regard to the endpoints in the clinical trial, we still have approaching the clinical trial. The use of AI can, and actually for some it's quite advanced, can assess digital endpoints like minimal residual disease, fibrosis, by the right tumor volume, trying to have, let's say, less variability around the trial site. What we are expecting in terms of what the regulators want to see. As I mentioned before, EMA expects consistency and reproducibility and validation against traditional outcome, such as PFS or overall survival. And this is particularly important that what it's looking when we look at some AI tools is the fact that this validation, this process of validation of AI should be oversight every time from expert or from human. This is the point. So it's completely useless to propose an AI analysis or an AI extraction without having a validation or without having human oversight of the process. But we will see later how this can be let's say exploited. So yes, the other question that I was thinking was about yes, we can improve patient recruitment using predictive AI or analysis or monitoring for example, for safety signal or efficacy, and then try to somehow also have an adaptive design in the study in terms of we can change or we can modulate by using AI. Obviously, it's important to understand from a regulatory perspective if the AI endpoints are generated, are derived from the use of AI, and actually the first question from a regulator is this endpoint validated? And how this validation has been performed. So there is the review of the training data used for the validation, and also how this validation take into consideration let's say the so-called outliers of the algorithm. And also how these methods, this approach, take into consideration, for example, the poor quality of missing data. These are, let's say, how regulators approach to AI data generated. Another point that I want to touch is which is again encompass the strategy. The strategy, as I told you, was to improve clinical trial, to use it as a safety signal setting, so in the setting of the pharmacovigilance. So when we talk about pharmacovigilance, it's quite easy to understand that if we use AI for analyze a large amount of data or signals, obviously it's quite easy to pick up signals, so also quite faster, I would say, because obviously there is a limit in the manual process. Obviously, this has a huge regulatory value because there is signal detection allow EMA and the Pharmacovigilance Committee to improve safety warning and all this stuff. This is a very important point in which EMA is currently investing a lot of energy. Here there are some example for with regard to the thrombotic microangiopathy, which could be, for example, used as as a pinpoint for picking safety signal. But actually, okay, what I mentioned is how the regulators are trying to approach the new field, are trying to approach the innovation of the AI put forward. But also, I want to say that we are embedded in the use of AI. And as you can see, we already use the large language model. Also at EMA, there is some use of AI, for example, in using and analyzing a large amount of data. Obviously, there are conditioning using AI, a language, and a large language model within EMA, which means that you should use in a closed environment in terms of the data should be closed to the external parties, to the external world. So the model will work only on data that you have feed the model. And this is obviously also, there are a series of restrictions. So yeah, no confidential data may be processed. On safe platform, as I mentioned, output must be verified by expert and compare with original data. This is one of the challenges that we have. So we need to have experts which analyze the output, but we also need experts in the field of the AI algorithms and so on. And obviously the impact is quite clear because the speed of the output is very important. We have different tools which are currently I touched during my previous slide, in which hematology, since we are speaking in a hematological field, can have benefit or can have a very, let's say, can have an impact on their day-by-day practices. For example, we have AI-driven tools for dosing, prediction on neutropenia, planning transfusion, for example, MDS. And obviously, EMA every time needs to can assess, and also have to assess if the use of these tools are, let's say, are adherence to the principle, so the validation, to the transparency, and to the fact that there is a human validation behind. And this actually it's let's say the leitmotif for what's happening now in the field of AI. I don't know how time. Okay. These are, I will leave obviously for the, maybe for later. There are many possibility and many way to explore the use of AI in dermatology. We talk about minimal residual disease, but there are also all other fields which are evolving. We already know for meta-analysis and so on. So the use of the AI tools in different settings, it's very important. What is important at the end of the slide is that every endpoints who derive for AI process should be validated in every protocol for EMA to make it acceptable from EMA, and also maybe to use for approval. As I show you at the first slide, in the 2025, there was the first qualified AI tools, which set a precedence, which is very important because to some extent has been recognized the utility or of the importance of the AI tools within the life cycle of our medicinal products. And actually, this also set the scene in terms of which are the data needed, which are the training, let's say the training data needs, which are the expert input, and how you validated this. So this is I would say one of the example which sets the scene for having qualification of AI tools. You already know what is going on with the AI tools. So when we talk about AI, we talk about something that is already embedded in our stuff. It's already embedded in our cytometry flows, in our NGS. So we already have program that, to some extent, apply an algorithm which extracts data and which put forward results to you. So you are already using AI. And actually, what is the added value is the fact that you are faster and you are actually more performing in terms of analyzing the huge amount of data. Since the timing is getting closer, I will approach to the final of my discussion, but I'm happy to have any question. So, which are the opportunities that we have? I'm highlighting for hematologists, but I think that all the researchers actually can have benefit from this kind of use. So we can have faster trial using AI-driven endpoints, for example. We can have and we already have real-time analysis from NGS data. We have integrated certification, for example, in MLMDS. We can have the improvement of safety signal, which actually is a very important point for the life cycle of the product. And also we can have some specific tools with regards transfusion, for example, and so on. Let's finish with something which is important. So which are the concepts to keep in mind? As you know, all the system, and particularly AI model, have an intrinsic bias. So AI tools may reflect these biases and amplify biases from the limiting training data and also for example, underrepresentation of some data. So what is important is to try to clarify the so-called black box in AI model. So what is important is to open this black box, try to give an explanation which means the human operation. So which means that the expert in the field, which means that the data scientist is able to understand how the inference from the algorithm has been generated and if this generation is consistent with regard to what is expected from the expert. And this is why it's important that the human and the clinical judgment overall. So this is let's say the major and the critical issue that we have in using the AI model and in analyzing AI model. We have to understand what is happening inside because because on the other hand, it would be very difficult to believe or to, let's say, assess data coming from the blue. This is to make it, but we can discuss. So what we need to remember, or what we need to keep in mind when we are approaching AI. So EMA expects that AI in clinical trial, in developing, in diagnosing, whatever, in monitoring, it is expected that these should be validated, should be presented in a transparent model with a clear human oversight of the process and also of the algorithm. And the question is what hematology can do. So what we can do is just to ask if the AI data generated are the data that we are expected, and moreover, we cannot substitute the outcome of an AI data without having the expert, let's say, confirmation of this. And then this means that we have to face this in the next year. It will be actually quite important. I'm coming to the end, and actually this is just to recall what Paolo was saying before. So the medicine network is doing an amazing job, is doing an amazing, let's say, interaction among all the competent authorities, because as I told you, EMA raises some guidelines, but actually each single competent authorities, such as IFA, is a single point in the network, which in the is able to react as well. And then what is saying Paul before, please contact the agency, try to have an early dialogue, especially if you want to qualify or if you want to explore new use of AI system or just for, I don't know, for accelerating enrollment in clinical trial. And obviously, I will stop here, and I'm happy to take a question afterwards, and just to tell you that this presentation has been made by AI with my supervision, obviously. Thank you very much.
Many thanks, Armando. We are perfectly on time. It's 12, and now it's my pleasure to give the floor to Saverio D'Amico, which is an engineer data scientist from Humanitas Research Hospital, and is surely an expert. And so he will discuss the challenges and the opportunities of AI. I'm very interesting and happy to hear you. Saverio, please.
Yeah, thank you so much for the kind introduction, and thank you for inviting me into this very interesting meeting and initiative today. Yeah, we talk about. I don't know if I am truly an AI expert, but I try to do my best, and with my team, try to use AI as a technology to answer some clinical questions and clinical issues and unmet need that are still open, in particular in rare disease context. So I'm going to share my screen. Can you see it also in presentation mode? I don't.
Oh, you interrupted.
Okay.
We see the. Okay, perfect.
Okay, great. So as I see it today, I want to discuss about opportunities and challenges of artificial intelligence, of course, in the hematological field, which is the field in which we have started in the past 5 years, and in our perspective for two main reasons. The first one is, of course, that we are working in this field, in particular with the collaboration with Professor Matteo Della Porta. But the second reason, the most technical and clinical one, is that in our perspective, hematology is the more complex field in which AI can be really an effective solution to solve some unmet needs. So in this environment, in this context, AI can be, let's say, developed in a very strong way in order to be in the future scalable for other clinical domain and for other clinical aspects. Before going, I would say, in the details regarding presentation, I would like to introduce the concept, the basic concept about artificial intelligence, that just to have a common, I would say, vocabulary to easily understand from any of us about all of the single applications. So right now, everybody knows about artificial intelligence, and everybody knows, of course, the concept and the possibilities about AI. But I would like to make some order in terms of context and terms, and starting to, let's say, see artificial intelligence like a matryoshka, so with different concept once we go deeply within it. So AI is basically the field of the study with all of the concept inside it. But then we have machine learning, as you know, is like some I would say a mathematical function of mathematical tool that try to understand some patterns from historical data and trying to reproduce it in the brand new data just to create prediction or classification or so on. Then we have the deep learning. Now everybody knows about deep learning, but it's basically the use of machine learning, but with a very innovative tool, which is the deep neural network, another mathematically function that try to simulate the work of a human neuron in order to enhance the power and the performance of machine learning, and in order to make, I would say, the use of unstructured data, for instance, images or text, audio, video, and so on. And then going deeply, the more deep scenario is the one we have nowadays. So the generative artificial intelligence, the technologies behind some AI tools like ChatGPT or Gemini or so on. Generative AI is the most right now innovative tools in order because basically it's so powerful, and there is a lot of, I would say, performance improvement with the respect of previous, I would say, traditional AI tools. And in particular, with this slide, I want to introduce you the description of some AI application once we go deeply with the definition of the complexity of the technology. For example, when we talk about artificial intelligence, of course, we have a general perspective. We can talk about in a general way about medical product, medical device, or medical software, or decision support system. But as I described, once we go deeply with the definition of the complexity of the methodology, we can, let's say, define more and more precise application. Regarding generative AI, we have the most innovative right now, and the most innovative one. Also, my colleague previously showed some of these technologies for clinical trial and for clinical endpoint, which is the generative artificial intelligence, and in particular with the clinical application about synthetic data generation, preventive care, and the new innovative one, digital twin technologies for therapy optimization or personalized treatment. Of course, before talking deeply with the AI application, I would like to stress the concept that using generative AI, which right now is the most innovative and performative technologies we have, considering artificial intelligence, does not mean to use ChatGPT or Copilot or other application. As you can see from this slide, in fact, ChatGPT is like a small point in the worldwide um, scenarios of generative AI tools. So use leveraging on generative AI technologies just does not mean, once again, to use ChatGPT, but to use the technologies behind this such of this product. Because in order to create a very applicable and performative and transparently reliable, of course, way of the use of technologies, we really need to understand the clinical need, the clinical met need, and the clinical problem that we'd like to solve with this kind of technologies. That's why apply, I would say, artificial intelligence in the healthcare field, in the healthcare sector is not easy, and in particular is not the same thing as applying the other industries or other sector. And this is particularly, I would say, evident from the perspective from the WHO, the World Health Organization, that in 20, more or less 5 years ago, in 2021, give us a sort of guidance and ethic guidelines about the ethical use and the validated use of AI in health. And these kind of guidelines can be, I would say, summarized into three main aspects that is shown in this slide. And this, like the first one, the models need to be transparent. So we need to build technologies in a more transparent way. So we need to understand why the model are taking such decision. And this is the concept behind entreprenability and explainable AI, of course. And my colleague talked about it in the previous presentation, that we need to build reliable models. So as all of the other clinical application and clinical study, we need to create, I would say, clinical study in which we can validate the use of these AI technologies like all of the other clinical studies. So we need to create a training cohort of patients, and then also external validation cohort of patients, in order to, I would say, demonstrate the usability of these AI tools. And last but not least, we need to consider technologies that from, I would say, from the design application of such technologies need to be to consider the protection of data and data sharing. So, for example, European Commission already identifies some technologies that by the design are solving these privacy and protection data sharing issues, like, and I will go deeply in the details in this presentation, but like the synthetic data generation or federating learning perspective and algorithms. So, in my perspective, our work in Humanitas, and I am a biomedical engineering within Humanitas team, leading the research and development team, and also with Train, which is the technological startup spin-off from Humanitas Center, are building in order to once again solve clinical problem with technological solutions. Our work basically has started from European, I would say, initiative like European Horizon 2020 project and consortia like Cynthia, Syntema, and Genomed. So within this context, within this environment of collaboration between research institutions, hospital and medtech companies, and in particularly within the European initiatives, we build a technological roadmap that not put the technology inside every project, but try to use technologies once again to solve the clinical problem behind such this project. And within this context, we identified some solutions that I'm going to show you in this presentation for data extraction, generation of clinical evidence with the synthetic data generation, or to use this technology for enhancing and solving most of the problem of the clinical trial, and then to use technology for building a personalized medicine approach with the construction of the so-called the digital twin. So talking about the AI application that we are right now using and building in the hematological field, I divided this kind of application in such in 6 points. First one is natural language processing system. So a sort of AI tool that try to understand the natural language text in order to solve the data availability problem. So everybody knows about these issues, and in particular in rare diseases. So right now we have this data that the 97% of the data within worldwide hospitals. So I'm not talking about Italy, and I'm talking about Europe. I'm talking about the worldwide hospital situation, and that is that the 97% of the data are in unstructured formats, so they are unused for research purposes. So how we can solve such this problem? Of course, we can build tool that try to automatically structure the data from a medical text report, for example, or we can use a tool like this one to capture all of the information starting from the clinical patient conversation during an outpatient visit, for example. And this is one of the practical example that we are actually have built, and we are, let's say, validating within our hospital and with our technologies. And it is called ambient AI, so a sort of AI application that transcribe all of the conversation between the medical and between the clinicians and the patient, and try to propose a sort of structured medical report that already have collected all of the, let's say, clinical information to be structured. And of course, with already clinical and individual data in a structured format. For example, the blood test or the information regarding treatment or regarding, I would say, the patient reported outcome during the visit. Of course, this is a more, I would say, a more big picture behind that, because once we can use this kind of AI tool to automatically extract and structure the medical patient conversation, we can use this to be connected into another, for example, repository of clinical product, and automatically identifies in real time if the patient are suitable for different clinical trial or critical product. So with this system, artificial intelligence can be an resolutive approach within the problem of structured data. Of course, we can use such these AI tools, not only to capture the voice, but in a, I would say, a retrospective way to structure the already collected and created medical text report in order to, let's say, structure in a mathematical way the information within CRM, CRF, or REDCap or other, I would say, structured tool to be stored the data. And thinking more, I would say, in a more general view, we can use this application not only for the medical, the clinical and outpatient visit during the hospital, but as a tool to remote monitoring the patient during the remote monitoring in home, in house, or so on. For example, to provide information regarding the patient adherence with respect to a treatment, or to, I would say, monitoring some adversarial event that can be, I would say, during some clinical product or clinical information. And another kind of application, the most, I would say, the most innovative one right now, in particular with clinical trial, are the synthetic data generation. So, as you probably know, synthetic data is more or less is an application of generative artificial intelligence, and this is basically artificial data generated by, in this case, an artificial intelligence model that mimicking all of the characteristics and relationship between the real data, but with preserving all of the privacy and GDPR aspect. This is why synthetic data are have mainly two big property. The first one is that synthetic data are totally anonymized data, and this is not from my side, but also the AI Act and recently the new law approved more or less in Italy three weeks ago are basically have said that synthetic data are considering anonymous data. And the second one is that synthetic data are a sort of structured data that we can condition with respect to the clinical question that we need to answer. And in particular, one of the most notable applications of synthetic data is in clinical trial, just to solve more of the problem regarding the patient's enrollment or the burden of patient participation in clinical trial, for example, for placebo in particular, control arm, and so on. So with synthetic data, we can create, we can generate more structure, and we can simulate an enrollment of patients with respect to answer to some specific endpoint, clinical endpoint, or so on, in order to create a more precise and more, I would say, with less percentage, less probability of failure of the study, but with a more clinical evidence that we can use to design a more better way the clinical study itself. And of course, we can reduce the number of patients to be enrolled, for example, for the synthetic control arm, because we can leverage on historical and retrospective data as a source of information to create this so-called synthetic data. And this is why synthetic data need a few patients, a few historical patients to be trained. And then with these, I would say, training data, training real patient data, we can then, of course, training the model, then we can ask the model to create whatever we want patients with the characteristic that we are interested of. For example, if we have, I would say, a cohort of patients, about 100 patients dividing into 70 female and 30 male patients, we can train our generative model, synthetic data model, with this information, and then we can later ask to this model to create, to generate more 200. So in a way to augmenting this kind of data, 200 data set, 200 patients, but this time 100 male and 100 female. So also using synthetic data to balance the cohort of patients, or I would say to create more suitable and more precise, I would say, enrollment scenarios in which we can absolutely use these technologies to identify the clinical endpoint and to solve some issues regarding the number of patients enrollment, of the problem regarding the lack of enrollment in a specific, I would say, patients class. Another, of course, important part of synthetic data is the validation approach. Right now, I would say is not anymore a technological problem. We have a lot of technologies that can create synthetic data, but the most important part is the validation of such this data. And this is why we and my team created this tool of validation, which we call SAFE framework, which can design considering the three main aspects from the WHO guidelines in the AI tool, and considering that, we build this framework to be answered with these three main metrics: the statistical fidelity, clinical utility, and privacy preservability. 3% that we can use not only to understand and to validate the course of the synthetic data generated, but also to understand if the real data used as a source of information to create synthetic data are, I would say, enough robust and enough with clinical information to be used as a source for synthetic data. And this is so important because in artificial intelligence and in general, it's true the principle, it's right the principle of the garbage in, garbage out. So if we don't have a very structured data, very clinically representative of the actual real situation, no way to create a very robust model, AI model that can easily solve and robust solve all of the clinical domain and the clinical questions more or less that we want to address. And this is an example of an AI tool, the one we build regarding synthetic data generation. Right now, we have such this platform that automatically connect with the in-house data repository and trying to create whatever we want synthetic data already validated using the AI framework, validation framework that I would say that I describe in later. And this is an example of some already implemented and already validated synthetic data generation approach in different therapeutic areas, from hematology, of course, which is our first case of study, and then scaling the technology into oncology, gynecology, fertility, and neurology. One example in the hematology context is a clinical trial that we're trying to reproduce using synthetic data as a control arm. And as you can see from this trial published in Journal of Clinical Oncology (CCI) in 2023, we was able to reproduce all of the clinical endpoint using a completely synthetic data control group, substituting the real data population. As you can see also from the I would say from the Kaplan-Meier curves, that are showing the real control group and the synthetic control group. The two curves are not, I would say, one on top of the other, but they are the same behavior. And this is why synthetic data are not the real data, the real patients, but in another way. They are totally different patients, totally different data, but with the same behavior of the real one. Another application also, of course, in hematology is the generation of the so-called multimodal longitudinal data. That's why, of course, in hematology, we need to not only generate the clinical data, but we need some genomic aspect, omics in general aspect, considering also RNA sequencing, NGS, and so on, but also a longitudinal information for reproducing in a very robust way the clinical endpoint in a clinical trial. That's why we build, of course, and validating, presenting last year during the ASH conference in San Diego, these platforms that are capable of generating not only the clinical information in a synthetic way, but all of the multimodal data, considering also histopathological images and transcriptomic, endomic, and genomic features. And this is another example that we use in hematological in the generation of histopathological images that we used for enhance the, I would say, performance of a clinical model. Talking about very briefly regarding the I would say the regulatory aspect. Of course, synthetic data are very innovative technologies, but right now in FDA, in US, in FDA, we only have three examples of clinical trial that obtaining the approval using synthetic data, and these are this one. But we also have a scientific alliance between the FDA, the scientific community, starting in 2023 with the use of synthetic data as a control arm for a no placebo initiative. There is a lot of work. This is an example of that behind the synthetic data using with the FDA within the environment of synthetic data. What is the situation in Europe right now? We don't have right now any, I would say, clinical trial obtaining the approval using synthetic data, but there is a lot of investment behind that, and in particular with the European initiative called Cynthia, we have more or less 23 millions of investment from the European Commission to 32 partners with the aim of understanding the use and validating the use of synthetic data in clinical trial in these six showing in this slide, these six clinical case from oncology, hematology, of course, but neurology and also diabetology. Another very briefly kind of application of the AI is in for imaging analysis, and we can use AI to make an images classification, object detection, and semantic segmentation. In hematology, we already have some results in using AI for the histopathological, I would say, information extraction using in fact AI to extract information from the histopathological images in order to one assess more diagnostic accuracy in myeloid neoplasm patients, but also to create some, I would say, linking and relationship between the histopathological feature, the morphological feature, and the genomic pattern of a single patient. Then in order to create at least 3 point, third point, a more precise, I would say, personalized risk assessment. And this is some example of the results, but the most important one is that using all of the clinical information from genomic, for clinical genomic karyotype, but also imaging, more we embed information, more we have more precise model in order to create more precise individual personalized risk assessment. And all of this is, let's say, possible using AI as a tool for implementing such disinformation. Very briefly, another application is the federating learning. Federating learning is a new ways, a new artificial intelligence way to, I would say, rethinking about the sharing of the data. And this is a technology also considering by the European Commission, the European level, to in particular in the healthcare domain, because it changed the paradigm of the sharing data. So everybody knows that we need to sharing data to create the more precise model possible, because once we can share all of the data available from all of the hospital, we can collect, create a central repository, and then we can try an AI model based on these overall data. But in the healthcare sector is basically impossible to, or more very difficult to share all of this data across different hospitals. So with federating learning, we can avoid the data sharing, but we can create a single AI model into single hospital, and then we can share the knowledge behind what the single model learned. And this is not, I would say, a future perspective or future technologies. This is the present, the reality. And right now, within this another European initiative, which is called Genomed for All, we right now have connected different hospitals showing this slide that right now is sharing, I would say, not data, but what the model learned from the data that remains within the hospital system. And to conclude this application, I want to conclude this intervention and my presentation. I want to, I would say, going deeply behind the concept of the digital twin. The digital twin, in our perspective, is a way to, I would say, simulate different scenarios in the healthcare sector for individual patients in order to provide clinicians more clinical evidence for taking more precise decision. And in our perspective, digital twin are more usable for personalized medicine, of course, and clinical trial. In clinical trial, in particular, with the generation of synthetic patients, as I described, but in personalized medicine, in order to create a tool which works with individual data for individual patients in order to answer and address the most important clinical questions, which is diagnosis, prognosis, or the evolution of disease, and then the treatment strategies in order to address the disease, in particular with individual data within individual patients. Right now, we are building this kind of model between a very big project that we won with an Italian grant from Minister of Health, the FISA grant, and we are building this kind of digital twin to address this clinical question in hematology using some AI technology technologies to solve the problem of data collection. Of course, to build a digital twin, we need a lot of data. So we are using, once again, some technologies, for example, synthetic data for learning or voice model or data extractor model to collect all of this data in order to build a very strong digital twin model to be used in the future as the core technologies and the engine to be used as a virtual assistance to provide to one the clinicians a way to interact with this model and to answer, for example, the most suitable therapy and treatment strategy to an individual patient for personalized medicine. And second, to provide the patients a way to interact with clinical and to, I would say, collect all of the a patient-reported outcome in order, in a cyclical way, in a cycle way, in order to have a more precise, personalized medicine. So thank you so much. I would conclude my intervention now, and once again, thank you for the kind invitation.
Many thanks, because it was really an interesting presentation. It gave us really a look on the landscape and the many potential uses. So it ends our presentations planned for this session. And while I'm checking whether questions pop up, perhaps I have one, because I am starting from you, Saverio, and of course, uh, synthetic data. I think it's really an amazing development that could really change the landscape. And of course, it's what we call as high regulatory impact. Once you have the potential to have a complete synthetic control arm, I think on the one hand it provides enormous opportunities. On the other hands, you can imagine that regulators are scared to rely on that. And probably I go back one step back, because we struggled over the years in accepting real world data because of problems of of data quality and completeness. And so the fears of the regulators might be on the fact, and you pointed out to that, and I'm happy to hear that perhaps if the starting data set is not that good, then you could even amplify, if you wish, the bias in synthetic data. So my question to you is, putting this concern together, what you showed in the other slides with regard to the fact that AI is also able, in a way, to perhaps to help in this regard. Uh, but also there are a lot of concerns. So how do you see this issue of data quality can be addressed and solved in the next future, maybe also using the AI in this regard to help? So my question is, because I really I'm in favor of this. I would like this to happen, but I think that really this is a field in which we need to work together. You had to convince regulators, and we had to try to convey you the messages. So sorry for the long question, but I think it's a very hot topic on the table we have.
Yes, no, thank you again. And this is a very good question. And this is basically, I would say, the direction we have use and choose once we start developing such technologies. In particular, because the concept of the real data behind the synthetic data are so important. And we worked about this synthetic data since 5 years, vertical in the field of healthcare, and in particular starting from the hematological context. This is more difficult considering that we have rare diseases, so few patients, so very few unstructured data. In order to use synthetic data to solve such problem, we really need to understand if the real data source are, I would say, feasible to create such synthetic data. And to answering your question, I would say that the important part relies on the validation approach. So that's why we created, I would say, this validation model that we call SAFE. And I would say that the effort that we put into the validation model, into the validation model, is more than the effort we put into the generative one. Because right now is not more anymore, I would say, technological problem. So there is no technological problem regarding generation state data, but it's a clinical validation one. For example, and I will provide a very straightforward, I would say, answer to this question. With the framework that I present, we can measure the quality of synthetic data into three main aspects: the statistical fidelity, clinical utility, and privacy preservability. But with these three metrics that are not we don't want to reach a threshold because we are using such these three metrics all together, but to have a trade-off between the answer that we need to be addressed. So, for example, if we want to use synthetic data into a clinical trial, we need a strong clinical utility. So we need to put all of the effort to, I would say, enhance the performance of the clinical utility metrics. And this is in particular true with this metric. We also really address the problem with the real data availability. For example, if we don't have a real data source, very robust or very, I would say, with a realistic clinical scenario, clinical representative of the real world data, using as a source for real data, we will have a very low clinical utility because synthetic data in general are, I would say, sort of an amplifier, as you also highlighted. So I'm an engineer. From my engineering perspective, synthetic data are an amplifier. So it's try to enhance the ratio between the signal and the noise. So if we have small signal, we can amplify the signal of this ratio. But if we don't have a very clear signal, we will amplify the noise. So we will going into, I would say, not good direction and no way to use synthetic data to enhance our clinical perspective. To address this problem, we build the validation model, and with the validation model, we can really assess if not only the synthetic data have good quality to solve this kind of problem, but also if the real data, the real source of data are enough and enough good and structured to create a reliable synthetic data approach. And our perspective, just to conclude this long answer, but in my perspective, there is no generalistic way to address synthetic data. We need to really understand the clinical question. For example, in a clinical trial, we need to address if we are using synthetic data to enhance, for example, number of patients to address the first endpoint. But we need to address what is the first endpoint. So it's a matter of number of patients, or it is a matter of we need more data to amplify the patient, etc. So once we identify the clinical question, we create synthetic data and we validate synthetic data to address this question. So there is no generalistic approach. We need to consider all of these, I would say, use of synthetic data in a vertical way and validated it for the particularly and specific use.
Okay, so what you mean is that the research question is the point it might be considered in order to understand whether the data we have on which we are going to generate synthetic data is fit for purpose in that we have the kind of completeness and representativeness of data that we need to answer the research question. I guess it is in principle, I could agree with this approach. Of course, it's not that simple as saying in few words, because it's open a lot of questions and different evaluation to be done, and a multidisciplinary evaluation to be done that pertains not just to doctors and data scientists but of course yes, I think that's but I do not want to I want to listen others. I see there's one hand. Antonella, please go ahead.
Yes, is a comment and a question, a consideration about on this, because the first question today, this morning, is how can we generate evidence through more innovative approach to accelerate drug development? This is possible with the external control arms we discussed this morning, with the real world data, with the synthetic data and probably in silico trial. But for synthetic data, it's necessary to aggregate historical data. So that first we need national disease registry, probably a support at national level, especially in rare disease, to collect the data in rare disease. Because for a regulatory perspective, I, I think about synthetic data and I want to know how these data are generated, how these data have been evaluated, how these data have been validated so that in general synthetic data should be used as a part of a pluralistic evidence strategy and for this reason a regulatory purpose case by case discussion should be advised early with the regulators but at national level we need collection data probably with the registry to collect the data to support synthetic data, if I understand. This is important aspect because especially in hemoglobinopathy, if I think the heterogeneity of this disease, if we have a national registry with some data collected, probably this could help the clinical trial and probably the risk model inside the great heterogeneity of this pathology. But start, we need to collect real data. Thank you.
Aurelio.
It was very amazing session. And I have two questions, one for Antonella. And the question is about hemoglobinopathy. We know that these are chronic disease, so the outcome that company choose are outcome that could be measured in short time. Ferritin for thalassemia and vaso-occlusive crisis for sickle cell disease. This is a bias of this disease in comparison with other malignancy, hematological malignancy disease. And this means that we cannot have any other outcome if we don't have, will not have longer trial to evaluate, because Antonella is right when she addresses the attention on the variability of vaso-occlusive crisis. But the other aspect is that we need, in other case, we need to have, for example, evaluation of liver failure because we have cirrhosis in sickle cell disease for vasculopathy. And the outcome of vasculopathy is not possible to measure, to determine in a short time. This is for Antonella. For you and for Professor Magrelli, I have a question. It is possible to validate a score, prognostic score, and to validate it different novel methodology. But my question is, because of thalassemia and sickle cell disease are rare disease, we have the opportunity to collect data only in retrospective way or on real life. Are these sufficient to make a validation of this data? Because there are different papers, several papers on prognostic score, but they are based on retrospective study. Is this possible or is this a bias?
Okay, I'll let Antonella respond first, and then I can go.
I know the question because I think about the drug about the sickle cell disease and the problem in relation to the endpoint for for example, a sickle cell disease. But as I mentioned today, for regulatory perspective, first problem is the population. The participant enrolled in the research study for regulatory perspective should reflect the population affected by the condition or those for whom the treatment or intervention is intended. So that when non-validated clinical endpoint or biomarkers is used fit for purpose for those specific question, we suggest as regulators to validate before being accepted. Probably the support of study in post-approval can help to collect the other sufficient data in chronic disease. But until now, vaso-occlusive crisis, VOC for sickle cell disease, is the most frequent endpoint sickle cell disease, and this is the problem because most of these patients not have problem only related to the VOC, but there is anemia, acute chest syndrome, stroke, pulmonary hypertension. Probably in this case, the use of alternative endpoint or biomarkers can help. For this reason, we suggest early collaboration with the scientific advice working party and the other national and European activity at EMA.
I fully agree with you, and there is another comment. There is a difference in treatment of sickle cell disease among different countries because blood support is more
Yeah.
present in our country than in United States and France. This means that the VOC frequency is lower in our country than in other. Okay.
Okay, and then I'll try to answer your question, and what we usually would expect is that is a validation based on prospective data. Then we can, of course, the qualification advice procedure is exactly thought to discuss what the best way forward might be. I would say that if you would like to think about a validation based on retrospective data, that should be done in a completely different setting, data set, that the one that was used to generate the first hypothesis. So I want to say that in some instances, validation based on retrospective data might be the only way forward, but then at least it should be done in a completely different and separated data set. Ideally, it should be done prospectively. It's a given. I don't know whether I answered, but I guess that this is the kind of typical things that are being discussed in a qualification advice procedure because, in a way, it's a case by case based in judgment and a discussion that needs to be based on.
As you know,
So,
it's very, very difficult to have a perspective study of
no, but I was
30
acknowledging
or
that.
40 years in this. The model, the model could be the myelodysplasia, but in the myelodysplasia they make a registry, international collaboration, and there is the outcome like acute leukemia, or that is a different model.
Yeah, but on the one hand, I cannot think that prospective validation is something completely impossible. It takes time. So sometimes it could be discussed whether a retrospective validation in a different data set could be something fit for taking conclusion. But I think that I'm not sure that it would be completely impossible. But I think that it's a very good question and a very good topic for discussion. I see that we are running out of time because it's 12:46, and I guess the next. So if there are no, I do not see other questions or hands raised. So if you agree, Aurelio, I would leave the floor to the next session, thanking all the colleagues and the panelists who took part to this session. And I think that Andrea is chairing the next session. Am I wrong?
Yeah. I'm sorry. Yeah.
Andrea, are you?
Okay, thank you. And so this is the third session, and we try to translate all of these approaches into practice, into the real world. We can start with the first slide. Now I share the screen. Takes some time.
Professor, we can see the screen, but not the slides.
I try again. And so this is my PowerPoint presentation.
If you prefer, I can share your slides for you and you just say next. Okay.
Can you see my first slide?
Can you just put the slide, the presentation mode, the slideshow?
Is it fine this way?
Not yet.
Full screen. Can I go ahead?
Okay, perfect.
Okay. And so this is the title of my presentation. This analysis was promoted by the Garden Alliance, and I thank them for that. And the subject is to study the cost of gene therapies, and this is a particularly controversial issue. And the main models that have been published so far relate the cost of gene therapies to disease prevalence. And so we have an important definition to discuss in the beginning, because this is an important premise, the definition of value-based pricing. Value-based pricing is a standard methodology for pricing ordinary drugs. And according to this method, according to this approach, the price is proportional to the clinical benefit generated in the whole population of treated patients. An important point is that in this case, there are hundreds of thousands of patients. The main point of controversy is that the value-based pricing algorithm is not applicable to orphan drugs. This is because orphan drugs deal with a very small population of patients, and so the approach, this is the standard approach for most drugs, is not applicable to orphan drugs. And so we try to give a look at the literature, and this is a report that we published in 2010 in the BMJ, and in this article we found a result that reflects common sense, in that the cost per patient of orphan drugs is inversely proportional of the estimated number of patients. And so there is an inverse relationship between the cost of the drug and the number of patients. And this inverse relationship has, of course, two main aspects. There is a qualitative aspect, which is the inverse nature of the relationship, and then there is the quantitative aspect, which is, as you see the mouse on the screen, it is translated in the best-fit equation, and so the inverse relationship model is included, is associated with the parameters of the model itself. And so we have a qualitative relationship and some quantitative parameters. This study referred to the Italian setting and included a total of 17 orphan drugs. And at that time, of course, gene therapies were not available. And so this relates to ordinary orphan drugs, even though there was the difference between orphan drugs on the one hand and drugs designed for ultra-rare diseases, which reflect the initial shape of the curve. And so now, in 2025, now we have tried to focus on gene therapies, and in fact, this presentation focuses on the pricing of gene therapies, and the information is updated to 2024. It is well established that there is this inverse correlation that we have seen before between disease prevalence and treatment cost. Now, the point is to understand how this inverse relationship applies to gene therapies. To do this analysis, we selected the United States as the reference country, simply because enough data on this issue are available mainly for the US setting, And most of this information was obtained from the website of the FDA administration. And so prevalence and incidence that you will see later on refer to the US population. And this is an important limitation as already a few minutes ago. But this is the only research that we could make based on the need of the information required for this analysis. The nonlinear regression model was confirmed. Also, this analysis focused on the United States. And as I pointed out before, we used US prices. And so after a standard search across the FDA website, we identified a total of 10 gene therapies. We selected only those gene therapies that are administered as one-shot administration. This selection is required because we need to have homogeneous data, and it is important that the administration is not on. This table shows the name of the 10 gene therapies, the clinical indication, the date of FDA approval, the incidence reported in the FDA HTA report, and it refers to the United States, and the price that has been agreed upon because these drugs have been marketed, have been introduced into the market. And so we cannot examine all agents one at a time. It would take too long. And I show the result of our analysis. Agents for hematological diseases are highlighted in red. You see that the number is quite small, but this is the total number of gene therapies that we have available in the United States. And the first important finding is that the inverse relationship published 10, 15 years ago for ordinary orphan drugs is confirmed also for gene therapies. The shape is somewhat different. There is not a steep increase of the curve in the first part of the curve. The curve tends to be more smooth than that we had seen before. And from a qualitative point of view, the model remains unchanged because this is an inverse relationship. From the quantitative point of view, as expected, the parameters are different. However, it is important to note that the correlation coefficient is sufficiently high, and from a statistical point of view, this relationship is statistically significant. And so, to be more accurate, this is the result of the statistical analysis where the correlation is significant and the p-value is below the threshold. And this is the best fit model, which includes essentially two fixed parameters, two parameters that are this empirical values, this proportionality value, and the exponential. And so there are only two parameters that are included in the model. The other parameters are the incidence of the disease and the result generated by this best fit model is the cost computed according to the equation and to the graph that we saw below. And so this is the graph. We have seen the equation, and this equation is intended to have a general validity with its limitations, of course. And so I would like to point out three observations on these preliminary results. The first observation is that there is a similar trend to the previous study. The previous study was limited to orphan drugs. Also, gene therapies show a very similar model. The difference is that the increase in the curve is steeper for ultra-rare disease, and this characteristic is more pronounced for the old analysis of orphan drugs and is less pronounced for the updated analysis based on gene therapies. And so this inverse relationship is confirmed, and the main driver of the result is the expected number of treated patients. This is the reason why the value-based approach is not applicable to orphan drugs and to gene therapies. This point is important because the fact that the value-based price is not applicable to orphan drugs has the consequence that the model that we can propose for orphan drugs is fully empirical. It has not a theoretical basis that includes the survival benefit, the improvement in quality of life, and so on, that are the parameters typically considered by the value-based approach. And so we need a separate model which has the disadvantage of being of empirical nature. And this is not a small limitation. We confirm that ultra-rare diseases deserve a higher price, and this is consistent with the analysis performed in 2010. And prevalence-based pricing is intended to reflect the market dynamics, because the investment in research is distributed across a small population of patients, and this is the reason why you cannot apply the value-based approach. Each patient deserves a higher cost than that determined by the value-based price. The main problem is that this increase in the price recognized to these agents is fully empirical. And there is no model proposed, no specific model proposed thus far in the literature. So this is surely a very preliminary model, but it is the first that has appeared in the literature. It warrants further research, can be useful in shaping future pricing strategies, but we think that this model should be initially applied to ultra-rare diseases. Probably it is premature to apply it to other orphan drugs. Other orphan drugs, as we saw in 2010, perhaps can be managed using value-based price or more common schemes or algorithms of drug pricing. The main controversy lies for ultra-rare diseases, and this is still an unsettled question. We need further research to advance knowledge in this area. Thank you for your attention.
Eh, Andrea?
Eccomi, here I am.
Yes, there is another speaker after you that you introduce.
Thank you. Professor Rivetti again.
Good morning, everybody. My name is Pia Rivetti. I work at the Italian Medicines Agency, like Paolo, Antonella, and Armando, who you listened to before. Today, I am going to talk to you about joint scientific consultations and joint clinical assessment and their impact on development and access for rare disease products. Can you see my slides moving?
Yes.
Okay, great. Joint scientific consultation and joint clinical assessment, JSC and JCAs, these are odd acronyms that are becoming more and more common since the entry into force of the so-called HTA regulation, which is the European regulation of December 2021 that defined a framework for joint HTA cooperation between member states and also in collaboration with the EMA. So this is the talk I'm going to give on the topic of these two tools that have been developed for joint HTA in Europe. So first of all, HTA it stands for Health Technology Assessment, and so a health technology is anything from a medicinal product to a medical device, in vitro diagnostics, but also procedures. And basically, what happens, so HTA is what is done normally at the national level. So after the authorization of a new health technology. So we're going to talk about medicinal products mostly today. So when at the national level, the government needs to decide for the national healthcare system if they want to use a certain new product and if they're going to reimburse it. And basically, the HTA is something that allows to assess if a new medicine is more effective than what is already available in a said country, in a specific healthcare system, and if the expected costs and benefit represent sufficient value for money if you compare to what you have already available. In general, it's not very different from what we daily do when we go shopping at the supermarket or we go shopping in normally we decide, we assess if something is needed, and then we decide if even if it is needed, if we want to buy it, so if it is good value for money. So HTA is basically a framework to do so for health technologies. And HTA has two domains. There's a clinical domain and a non-clinical domain. The clinical domain deals with the relative safety and efficacy, which is also the effectiveness. So how the drug is going to actually work in the population in that specific country, considering the specific alternatives that are available in that country. And then you have the non-clinical domains, which are pertaining the economic evaluation, the ethical, organizational, social, and legal aspects that can come with the introduction of a new health technology. And so far, this is the access pathway in Italy. Well, it has been like this until the beginning of this year. In which HTA and pricing and reimbursement was performed by AIFA, by the Italian Medicines Agency. So a product had an initial phase of research and development with the possibility of having a scientific advice, as we were informed of earlier in the previous session. And the scientific advice aims at helping developers to design the right trials in order to have more streamlined and successful evaluation by EMA for European assessment and granting of a marketing authorization. After that, if the marketing authorization holder wants to be reimbursed, wants their product to be reimbursed in Italy, they will have to submit a dossier to AIFA, and AIFA will do an HTA assessment. So it will compare this product to what else is already available and reimbursed in Italy. And then there's going to be a negotiation for definition of pricing and reimbursement. And after that, then there's going to be the regional organization. So everything that happens after the decision of the AIFA. And now that we have this new HTA regulation, we have these two tools that are aiming at doing HTA at the European level. So we have the joint scientific consultation that happen very early during development, during research and development, and they can happen also in parallel with the scientific advice, which means that the developers can ask for advice not only to EMA, but also to the HTA coordination group in order to have an advice on a design of a study that will be successful not only in terms of regulatory needs, but also in terms of HTA needs. And afterwards, once the product is submitted to EMA for evaluation, starts the concomitant JCA, which stands for the Joint Clinical Assessment. So it's going to be a joint HTA assessment that happens at European level. So this is the framework that has been introduced by the regulation. And this is going to introduce a real European HTA, and the need of a European HTA, of course, historically comes from the fact that the European Union is not, there's a huge variability between the different countries, and not all European countries have HTA bodies, not all of them use HTA bodies in the same way. HTA bodies can do the assessment, the clinical assessment, or the reimbursement decision, or the pricing definition. It does not happen in the same body. It does not happen in the same way in different countries. And all of this, and also there's a difference also between medicinal products and medical devices. And all of this variability means that the same product can be assessed very differently across different countries. And this, of course, is a problem for companies because they are not sure how, where, and when they will be able to market an authorized product in the European Union. It is, of course, a problem for patients as well because there's not going to be the same access across the European Union. There's also a problem of workload because there's a huge duplication of dossiers and evaluation. If you think that the same company will need to prepare, in theory, 27 different dossiers, and that 27 HTA bodies across Europe are going to make a similar evaluation of the same dossier. And of course, with a great variability in outcomes. And then there's also an issue of time to patient access variability. In the graph below, you see the red triangles are showing the average time between the decision from the European Commission on the marketing authorization and the time in which a centralized product is actually marketed in all these countries. And you can see that it ranges from less than 5 months up to almost 50 months. And so this, of course, should be addressed somehow. And this is basically all of these are the aims of the HTA regulation and of having a European HTA, which is not a new thing because since 2006 the UNETA initiative has been aiming at doing European HTA based on a voluntary cooperation. And the aim of this initiative was to assess the new therapies jointly across member states, but also to share the expertise and, in fact, avoid the duplication. So if you want, this new regulation is the culmination of the UNETA initiative, and the aim is to increase equality in access, the efficiency of the HTA assessment, its predictability, and of course this should call more investments in Europe. So this is the framework that we are talking about, and the regulation came into force in 2021 and was actually applied starting from January 12th, 2025. And it's starting with a staggered approach. So up here you see that the EMA has given 114 positive opinions in 2024, and since this is a huge turnaround, there's a huge turnaround in EMA, but of course it's also an agency that has been working for almost 20 years. The HTA regulation sees that the HTA for centralized product is going to start gradually. And right now, from 2025 to 2028, we're going to have mandatory JCA for cancer medicines and advanced therapies only. And then from 2028, we're going to have mandatory JCA also for orphan products. And from 2030, we're going to have mandatory JCA for all centralized medicinal products. So this is the plan, and actually, of course, there's huge variability from what is expected to then what actually happens. For instance, for 2025, we were expecting 17 new oncologicals and 8 new advanced therapies, but in fact, we only received 8 new oncologicals and 1 advanced therapies that are currently in evaluation both by EMA and by the HTA coordination group. So we'll see if this, this should This should only be a delay. Perhaps the companies are delaying the moment in which they are submitting because they need to understand how the new JCA procedure is going to work. So the governance for JCA and JSC is described here. So it is the European Commission that organizes the whole thing. The European Commission is giving support, technical support, administrative support through the Secretariat. And they are supporting the Member State Coordination Group, which is composed of the representatives of all 27 member states. And this coordination group has several subgroups that are also composed of representatives from all 27 member states. And they have specific tasks. So there's going to be a subgroup that is going to take care of joint clinical assessment. Another subgroup is going to take care of the joint scientific consultations. And there are two more subgroups that are more transversal. One is taking care of methodology and drafting guidance documents. And the other one is working on horizon scanning and defining and identifying emerging health technologies. And all of these interact with a stakeholder network so that there's some feedback coming from stakeholders on how things are going, what is needed, and so on and so forth. So this is not a closed circuit. So now I'm going to go a little bit more into the detail of these procedures. The one that is actually mandatory is the joint clinical assessment. So basically, starting from January 2025, when a product so now it's only for new oncological and advanced therapies, but from 2030 it's going to be to all products that are asking for a centralized marketing authorization. On day zero, when the procedure is submitted, when the dossier is submitted to EMA, there's also going to be a part of that dossier, basically only the clinical overview, that is going to be sent to the HTA coordination group and in particular to the JCA subgroup. So this is starting at day zero. And then the subgroup is going to have 100 days to do the scoping. And the scoping is a process in which they're going to define what is going to be the aim of the joint clinical assessment. What do we want to know about this new product? And then once the scoping is done at day 100, the procedure will go back to the developer that will have another 100 days to prepare the actual dossier in which they're going to show the data that were requested by the subgroup. Then there's going to be the drafting of the actual report by the subgroup. And in theory, only 30 days after the decision from the European Commission on a marketing authorization, so after the positive opinion from the CHMP and the final European Commission decision, we have 30 more days, and then the JCA report should be available. So this is the plan. We have not done any JCA reports yet. We have procedures that are ongoing, but none is at the state of publication of the JCA report. So hopefully the timelines are going to be exactly like they were thought by the drafting group. One important thing to keep in mind is that the joint clinical assessment is only relative to clinical HTA domains. So it is going to discuss the benefits compared to existing treatments in terms of safety and efficacy. And it is therefore going to be a scientific analysis of a health technology with information on relative effects, the degree of certainty of these relative effects, and the strength and limitations of the evidence that was supplied by the developer or marketing authorization holder. On the contrary, the non-clinical assessment, so the parts on economics, pricing, are completely in the national remit. So there's not going to be any recommendation on added value, place in therapy, or reimbursability in the joint clinical assessment report, because that will be, in any case, evaluated nationally. So I want to give a little more detail on the scoping phase, because over the months I realized that there is some confusion on how this is working. So as I was mentioning, when you have a new health technology submitted to EMA, the HTA coordination group will receive information on the claimed indication and on the clinical overview. And from that, and in theory, in a completely not data-driven fashion, so in theory, you would just need the claimed indication, the subgroup will define the PICOs. The PICOs are basically each member state is going to see in their country which is the patient population that would be corresponding to that claim indication. Is the claim indication corresponding to a population that is suitable in every country, or should that population be splitted? Because, for instance, there's a difference in treatment between pediatric patients and adult patients, or some mutations are considered in a country because there's a specific treatment, but not in other countries. So the patient population needs to be decided on. The intervention, of course, is going to be the health technology in discussion, and the comparison is going to be all the treatments that are available in the claimed indication population in every country, and then also the outcomes of interest. So this means that during the 100 days of scoping process, there's going to be 27 plus Norway, so 28 assessments that are going to make as many PICOs as needed. So it could be only one for each member state, and they could be all the same in all 28 member states, or not, because there will be differences, especially in the comparators. And the work that the assessor and co-assessors, so the member states that are chosen to lead the procedure, is basically to merge all the requests from the different member states without leaving anything out, which means that there's not going to be, for instance, the PICO of Italy, or that there's a negotiation between member states on which PICO to keep or not to keep, but there might be a merge so that the Italian PICO might be the same as the French and as the Swedish PICO. And so it's not going to be identifiable anymore in that way. But all the information that is needed by all member states is always kept. This means that there's going to be a list of PICOs that could be 1, 2, 7, 10, 15, or 20 PICOs that represent all member state requests and needs for their national assessment that are going to be sent to the developer. And the developer will need to reply to those PICOs with data coming either from randomized control trials or from indirect comparisons that will be done between different arms or different trials. They can take information from registries, from observational studies, and so on and so forth. And during the drafting of the report, assessor and co-assessors, with the help of member states, will assess how the quality of this evidence. Can we rely on this evidence? Is it good enough to then inform national decisions on pricing and reimbursement? And an important thing is that in all procedures, patient representatives and expert clinicians are involved in a mandatory fashion. So for each procedure, there's a selection of patients and clinicians that are experts on this, on each pathology, so that they can also give the view coming from, clinicians and patients in all procedures. So the other tool that I was mentioning is the joint scientific consultation, and this is a tool that is was conceived to obtain guidance on data analysis and everything that could be necessary in order to facilitate the generation of evidence so that it meets the requirements of a JCA. So it is really it goes in parallel with the scientific advice. It is exactly the same kind of tool, but it is declined in an HTA perspective. So who can do joint scientific consultation? We have eligibility criteria for this. So there's also a great limitation on the number of joint scientific consultations that are possible to be done now, which is why we need eligibility criteria, but also selection criteria. And hopefully with capacity building, we're going to be able to work like the Scientific Advice Working Party. But so far, this year, we had 7 procedures, 7 health technologies that were assessed. And tomorrow, well, then I think on Monday, there's going to be the publication of the 2026 program. So we're going to know how many joint scientific consultations are going to be done in 2026. And this is why we need eligibility criteria. And the first one is that the procedure, the health technology, needs to be likely for a JCA. So if it's an orphan product, it should be considered that it will not have the EMA submission before 2028, for instance. And of course, the pivotal study must not be started because otherwise there's no point in doing a joint scientific consultation. And then we have selection criteria because if we receive many requests, we need to prioritize them. And the selection criteria are the unmet medical need, if the product is the first in class, what is the expected impact on patients or national healthcare systems, and also if there's a specific relevance for Europe, or if the health technology belongs to the fields of research priorities in Europe. And what is going to be asked by the health technology developer in joint scientific consultation are all aspects related to study design. So aspects on the population, on the interventions, on the health technology per se, on the comparators that are that make sense for that specific technology in terms of HTA, and of course the outcomes also considered in terms of HTA. And there's also the possibility to have replies and comments on health economics, so modeling for health economics, and in general non-clinical domains. But since this is not a mandatory part, because not all member states have HTA bodies that have health economics in their remit. But I mean, there's also possibility to ask health economics questions. So to help the developer to define which are the right assumptions for their models. As I was mentioning earlier, we had only 7 slots this year on 2 requests periods, because in 2025 we did only 2. But from next year, we're going to have 3 request periods. And that is a period of 1 month in which health technology developers can send their requests for joint scientific consultations. And then over 2 weeks' time, the subgroup will do the prioritization and decide which procedures are to be done in the following months. And a very important thing to remember is that joint scientific consultation can actually be done together with the scientific advice. And this is called a parallel procedure. In a parallel procedure, you're going to have a perfect alignment in timelines between scientific advice and joint scientific consultation, which allows the health technology developer to have at once the comments from regulators and from HTA experts. And like the scientific advice, it is not binding. So the comments that come from the subgroup are not binding, but deviation must be justified because, of course, if you ask for an advice and then you don't follow it, you should explain why that was not done or not feasible. So this is the timeline of the procedure. It's the same timeline if it is in parallel with the scientific advice or not. We have always a discussion meeting at around day 60 of the procedure, in which there's a direct meeting between the health technology developer and the subgroup. And in case of a parallel procedure, there will be also the scientific advice working party there. And we also have the involvement of patients and clinicians, both in the initial parts of the procedure and present at the discussion meeting, which has proven extremely helpful because they give an extra insight, other from the regulatory of HTA perspective, but on the real world perspective, on the feasibility of some things that are proposed by the developer or proposed by regulators and HTA. And then at the end of the procedure, if it's a parallel advice, the developer will get the advice letter from the Scientific Advice Working Party and an outcome document from the Joint Scientific Consultation Subgroup. Or if it's an only JSC procedure, then it's going to have exactly the same timeline and function, but there will be no discussion meeting with the SOP, but only with the HDD and clinicians and patients, and there will be only one outcome document from the Joint Scientific Consultation Subgroup. So in conclusion, the take-home message is that especially for advanced therapies, rare diseases, so we are not in scope yet for orphan products for JCA, but they are already in scope for joint scientific consultations. We often see the problem of single-arm trials, low patient numbers, and also short follow-up, and the use of surrogate endpoints that are due to the high unmet need and the want to go quickly to the patients. But this is translating in a huge difficulty in defining added value on a national level from an HTA perspective, because things that can be sufficient from a regulatory point of view for an absolute benefit risk assessment may not be enough for defining an added value with respect to something when you do HTA. And this difficulty is responsible for a limited or even a staggered access of orphan products, and we see that they are not, we know that they are not widely available in all of Europe. So hopefully the HTA regulation with these new tools, and perhaps especially the joint scientific consultations for these type of products, are going to allow an active interaction between the developer, the regulators, and the HTA, and therefore increase the research plan acceptability and the research quality. And this, at the end, should allow to have more evidence-based and timely decisions on these new health technologies, and therefore a higher and faster access in Europe. And that's all. I thank you for listening to me.
Thank you. Is Andrea here? Is Andrea here? There is a question for you, Professor Rivetto, in a patient, Dario Martino. And Dario Martino asked if, according your personal opinion, we could think that in ideal future, there could be a more centralized negotiation for some specific innovative treatment, and HTA may help on this. I would like to add that in Europe we had the left of Bluebird Bio company in August 2021 because it was not negotiated the price of an antivirus procedure in Germany, where the incidence of the prevalence of thalassemia patients is very low. I think that another problem of innovative treatment is the heterogeneity in the incidence and for example, for thalassemia and sickle cell disease, that could be change the perception from the regulatory agency. What is your opinion about this?
Well, yeah, for sure, it's a very complex field, and there are many things that that play a role. In the case of Bluebird Bio, for instance, the IFA had negotiated the product, and we do have a higher incidence of thalassemia than Germany. But that was not sufficient. I mean, they were making a general European market consideration when they withdrew. So that, of course, is a huge issue. But imagining a centralized negotiation is currently impossible because that would mean taking in consideration the pricing of the drugs in all different countries. And we know there is a huge difference across countries. There are confidential discounts. There are different ways of allowing market access. You can have managed entry agreements, normal discounts, or limitations on the number of patients. And that also has an effect is also due to the incidence, as you were mentioning before, because there might be some countries that are interested in having a specific product while others are not, because they're not they do not have that kind of disease to such a prevalence that needs reimbursement in that case. So I think it is very difficult to imagine today a central negotiation. The systems are extremely different in Europe, and we know that Germany has a very different approach than all other countries. And we can also hardly imagine Germany deciding to have another type of negotiation, right? So to have a negotiation before access. Yeah, so I know that is also something that is worrying many people to have centralized negotiations so that somebody else is going to decide the prices that are going to be valid nationally. But currently, currently, I do not foresee this possibility unless there's major regulation overhaul.
Yes, I understand. But we live in the world where, for example, thalassemia patients in the United States have access to lentivirus therapy and not in Europe. That, that is very paradoxical, I think. It's very interesting, this the model of ATH. I would like to ask you, for example, population, with this one of the finding that you consider, population could be even the incidence or prevalence of the disease that you have to consider in your HTA model. The other question is the outcome of interest. Is there some difference between innovative treatment that could increase survival in comparison with innovative treatment that it could cure the disease? And in terms of cost evaluation, which is the comparison between the conventional cost of the treatment in a population with the median age in comparison with the cost of innovative treatment for the cure?
Well, so in terms of the PICO assessment, the population that is defined does not really take in consideration difference in prevalence. I mean, for instance, if you have the majority of patients that have a certain mutation, and then you have only a very slight population that has one specific characteristic, that population might not be requested in the PICO, because we know that most likely the company will not have the data. But in theory, as I was mentioning, the PICO procedure, the PICO scoping should not be data driven. So it starts from the claimed population. And then later on, we can go and check what is actually the data that the company has retrieved and see if there might be some missing information. But that is something that happens later on. So the general idea currently is to ask the PICOs that are necessary to understand the added value of that product in the whole population that is requested by the applicant, and if needed, to divide this population in subpopulations, and those will be all different PICOs. And then we will see later on, if there is no data, what will we do with that missing data? How bad is it that they didn't have the data? And of course, one important thing to keep in mind is that currently companies are asking for indications from a regulatory perspective, because they know that at the EMA, the application can change over time, because the EMA can restrict the population depending the indication, depending on the data that have been shown by the clinical trials. But this procedure is not really foreseen in the HTA. It is the initial indication that is the one that will go through the whole procedure. And if there is a major change in the indication, then there will be a delay in the generation of JCA report. So I would expect on the long run that the developers will change their strategy on defining the claimed indication and request a more data adherent indication. But we will see if that happens. And in terms of your questions on cost evaluation, this is not something that is assessed at all in the joint clinical assessment. It is something that is completely in the remit of national HTA bodies. And the only moment in which one can discuss cost evaluation is when, in the joint scientific consultation, developers ask for information on their models. But we are so far away from the moment in which they will have data, because they are in the planning phase of the pivotal study that will inform the model for cost evaluation. So it's really only some pointers that are given to the company. You should consider this type of endpoint for your model instead of that other endpoint, or there's difficulties with this model design. You could use another one. It is very general information so far, because we are so distant in time and we do not have the data to discuss the actual modeling.
Thank you very much. Thank you.
You're welcome.
Okay, we are moving to the end. If there is no other question, any other question from speaker from the no. Thank you again. Thank you again for your very.
Thanks for having me.
Okay. Okay, I have the last part of this very interesting webinar, and my duty is to the first is the announcement of the winner of the D'Agostino Prize. Garden organized this prize in memory of Dr. D'Agostino, that was a very innovative health director at Ospedale Vincenzo Cervello in Palermo. And I am happy to communicate, to inform you that the prize for this year is it was attributed to Dario Balestra. Dario Balestra works at the Department of Life Science and Biotechnology, and the prize was decided by the scientific committee that is constituted by Professor Angelucci, Masalam, Dr. Masalam, and Dr. Srivastava, and myself. And the prize was given because Dr. Balestra performed research on the use of base editing and prime editing for correcting the expression of factor 8, and this research showed us he obtained success in 20%, up to 20% restoration of factor 8, and this was the first study to provide the first experimental evidence of base editing and prime editing efficacy in reverting factor 8 mutation. And this was already showed in thalassemia and in sickle cell disease. So these are our projects for the next year. We are working to organize in March 2026 a webinar on the role of artificial intelligence in the development of data collection and prognostic score in rare hematological disease. This issue was discussed even today. And in June 2026, a webinar on how to improve diagnosis and management on sickle cell disease worldwide. And in October 2026, a webinar on gene therapy in the real life of rare hematological disease. I would like to thank again everybody, and I hope to have you in the next webinar. Thank you, and nice weekend for all.
Thank you. Bye.
Okay.
Thank you.
Thank you
Bye.
very much. Bye bye.
Bye bye.
Bye.